What a beautiful day we have today. I'd like to welcome each of you to our 111th Annual Plant Science Day at Lockwood Farm. I am Vicki Bamba Lewandowski. I'm the Information Officer here at the Connecticut Agricultural Experiment Station. Please note, we have a registration tent in between the barns if you're interested in getting on our mailing list and updating your information. The programs and agenda for the day are located at the registration tent. Please fill out our evaluation form and return to the registration desk on your way out. We do appreciate your feedback, and we do look at every single one of them. I would like to request the following for our benefit of our speakers today. Questions will be taken at the end of the talk when it's concluded. Please turn your cell phone off or on a quiet setting. And I thank you for your attentiveness, and we'll proceed with the program. First, I would like to introduce our director, Dr. Jason White. He will be presenting the morning greeting and opening remarks for the day.
Well, good morning, and welcome to, as Vicki said, our 111th Plant Science Day. The 110th was obviously last year, but it was a virtual event, which meant it was all online. So there were only 2 people at the farm that day, myself and Terri Jones, and we sat at a table right there. But as if the pandemic didn't complicate things enough, it was actually the day after Tropical Storm Isaias, and 7,000 residences in Connecticut had no power. Which is not an ideal situation for a remote plant science day. So we're happy, and as we say, welcome back. I do want to recognize some people who are with us today. Hopefully they're on their way here. But first is Representative Pat Dillon from New Haven. She'll be joining us today. And also Representative Dorinda Borer, who's from West Haven. So you can find them later today. Also with us is the Dean of the College, and I got to get it correct, it's always Caner, College of Ag, Health, and Natural Resources. Dean Indrajit Chaubey is with us. He's a very good friend of the experiment station. We work very closely together. So I just have a few comments, 8 or 9 PowerPoint slides, and then we'll move right into our first speaker for the day. So here is just a snapshot of our agenda that you have in your program. So after the morning greeting, we'll have a lecture by one of our scientists on beech leaf disease. And then we will have a few presentations, the Outstanding Young Farmer and the Century Farm Award. Unfortunately, neither of those awardees could join us today. So we'll be accepting those awards on their behalf. And then we have comments from the Vice President of our Board of Control, Terri Jones, as well as the Experiment Station Associates President. And also we have a special guest showing up around 11 o'clock. The Lieutenant Governor will be joining us. You all see her because she'll be driving up and parking right there. And then the rest of the day, and then our Century Farm Award presentation, which will be Brent Peterkin of Gather New Haven. And then the rest of the day is listed here. So as you look through your program, what you can see is that there are 51 experimental plots that are spread across the farm. There's an abstract associated with each one. We also have our barn exhibits, which are right behind you, and one for each academic department, administrative department. So we have entomology, the Valley Laboratory, plant pathology, environmental sciences, and analytical chemistry. All of the field plots and the barn exhibits are staffed, so they're there to answer questions and describe the research that they're doing. And we also have 18 outside exhibitors, which are spread about in that area. So in terms of an update, obviously the major update is I'm not Ted Andriatis. Ted actually retired as director April 1st of 2020 after 40 years of state service. We were hoping he would make it here today, but he did not. So I was actually appointed by the Board of Control as director April 1st, 2020. Although I've worked at the experiment station since 1997, actually I started as a postdoctoral research scientist and have been here ever since. My training's in environmental toxicology. I ran our analytical chemistry group for about 10.5 years, and then was vice director until I took over as director. Other major updates, obviously COVID-19 has greatly impacted agency operations since the beginning of 2020, and it continues to do so, obviously, till this day. In March of 2020, actually about 75% of our staff was sent home to work from home. The agency never closed. We were always open. All of our public health and diagnostic offices remained active. The tick testing program, the mosquito surveillance program, all of that ran through the pandemic. You were able to drop off diagnostic samples. Obviously, we had to dramatically change how we did things in terms of social distancing and masking, and a lot of that still remains in place in one form or another. About June 2020, we transitioned to 50%, and July 1st of this year, we were actually back at 100%, although we have a number of people who've requested to telework when they can, so we still have that in place. Other major updates, we have two major renovation projects that are underway. The first major one is that we have gotten approval to do a $17.8 million renovation and new construction on our Valley Laboratory, which is up in Windsor. The structure that was there, which is pictured on the left, was actually built in 1940 and has not been updated since, so this is an important project. But it also coincides with the 100-year anniversary of the Valley Laboratory, which was built as the Tobacco Research Station in 1921. So we're having a 100-year celebration on September 2nd. We can give you more details about that. That'll be at Windsor facility, and we hope to have a ceremonial groundbreaking for that project then. And then also a project on our New Haven campus. Many of our greenhouses were constructed in the 1950s, so we have funding to update and renovate a series of those, make them state-of-the-art facilities. And that project's actually out to bid right now, so we hope to start on that before the end of the calendar year. A number of retirements have occurred, and I actually went back through 2020 because obviously we weren't in person last year. But as I mentioned, Ted Andreatis retired. Frank Ferrandino retired in 2020 as well. This year in 2021, Dr. Abigail Maynard has retired. Peter Thiel, who was in our plant pathology and ecology department. Jane Kanappa Morrison, who was at the Valley Lab, and also Michael McKill, who worked here at the farm. We also have made a number of hires in the last year and a half. Dr. Susanna Cario, whose plot is over there in the center, whose poster. She was hired as an assistant scientist in our Department of Forestry and Horticulture. Christian Dimka was hired as the head of our Department of Analytical Chemistry. When I took over as director, he was hired to take over the chemistry department. Catherine Dugas was hired as a research technician in our insect inquiry office. Noel Khalil was hired as a research technician in the tick testing program. Jamie Cantoni was hired in a program within the Department of Entomology. And Ethan Payne was hired as a technician in our Valley Laboratory. Then we have some upcoming hires. We're actually have been in the process of hiring 3 new scientist positions. The first we just actually finished off. So Dr. Lee Winninghill was hired as a specialist in urban agriculture and food security. She's currently an assistant professor at Kentucky State University, but will be starting within our Department of Forestry and Horticulture first week in October. And then we have 2 other positions that we're currently conducting interviews for. One is on agriculture and the implications of agriculture in the environment. That position will be in our Department of Environmental Sciences, and we have on-site interviews scheduled in September for 3 candidates for that. And then our analytical chemist position is within our analytical chemistry department, and we're doing on-site interviews actually this month for that one. So both of those positions will be filled soon. And then the other major update is the adult use marijuana program. Our Department of Analytical Chemistry will be the regulatory testing laboratory for that program. As many of you know, that was legalized July 1st. Adult use cannabis was legalized. The program is actually being run out of the Department of Consumer Protection Division of Drug Control, but we will be their testing laboratory. Unlike the hemp program, where we actually test grower samples, our role in adult use cannabis will be, we're the lab that tests the private labs and also helps oversee the program. But importantly, that will come with three positions, and a scientist, a technician, and a quality manager position, all which we'll be filling before October 1st. So that's the end of my comments. I'm happy to take any questions if you have any. Or if not, we can move right to our first speaker. Well, again, thank you all for coming. Happily, the weather is cooperating. I hope everyone has a wonderful day, and certainly feel free to find me at any point if you want to have a chat.
For our first short talk of the day is Dr. Robert Marra. Dr. Robert Marra received his BS in biology and Russian from the State University of New York at Binghamton and his PhD in plant pathology from Cornell University. Following postdoctoral research positions at the University of Arizona and Duke University, he joined the experiment station in 2004. His research encompasses various aspects of tree and plant diseases of urban and natural ecosystems, focusing on the population genetics, ecology, and dispersal dynamics of plant pathogens. Current research centers on beech leaf disease, specifically the movement and spread of beech leaves nematode. Dr. Marra recently developed a methodology using sonic and electrical resistance tomography and non-destructively measure the amount of carbon sequestered in forest trees in order to investigate the role of internal decay in forest trees play in carbon cycling and the mitigation of global warming through carbon sequestration. Additionally, Dr. Marra maintains a long-term beech leaf disease monitoring plots distributed throughout the state and investigates possible occurrences of oak wilt. Dr. Marra?
Okay, so pointer is...
There's one there, or there's one here.
Do you want to adjust that display?
Oh yeah, this needs to be on full screen. Can everybody hear me in the back? Can anybody not hear me in the back? No hands, great, thanks. That looks good. Okay, so today I'm going to be talking to you about a disease that probably many of you, I hope, have seen by now, because it's a very easy disease to see in the forest or in the field. It's new to Connecticut as of 2019, as far as we know. So beech leaf disease was first identified in Ohio in 2012, and it was a mystery. I remember when in our various newsletters and that sort of thing, our fellow forest pathologists in Ohio were really curious. It was the strangest looking thing. And it took a while to really figure out what was going on. So I'm going to describe to you a little bit of the history of how this disease, we were able to determine, or scientists were able to determine the cause of the disease. It affects, as far as we know, just American, European, and Oriental beeches. Our native American beech, Fagus grandifolia, and the ornamental beech, Fagus sylvatica, and Fagus orientalis, which we don't see very much here. It's almost I would call it a congener of Fagus sylvatica, very closely related. Some botanists consider them to be the same species. It occurs in very, very far eastern Europe and the western edge of Asia. And so you're familiar with the European beech cultivars. You probably can't see my pointer here, so But these are, the copper beech is an iconic tree of city parks and cemeteries. Columnar beeches are popular in landscaping design, and as well the wilting or weeping beech is also a very popular cultivar. So those are all European beeches. American beech is what we see in our forests and in some people's backyards. And it has that really classic light gray bark. As you saw, well, in this picture here, the light gray bark, very, very characteristic. And also, as you can see in this picture here, the very distinctive translucence of the foliage. When you look up into a canopy, you can make out beech trees almost instantly because of that particular color and the light that comes through. It's very important in forest ecosystems for that reason, because of the filtering of light that it provides to the understory. So beech leaf disease, the progression of the disease as it's been learned since 2012 and as we've been seeing it in Connecticut now since 2019, is that in the early season, upon leaf emergence, as soon as buds break and leaves emerge, they emerge fully symptomatic. All the symptoms that you see in spring upon leaf emergence are the only symptoms that you're going to see for the rest of the season. And those symptoms are characterized by, if you can see in these pictures here or in the monitors, that dark banding. As one person at the experiment station here described it, I think he called it crazy Martian striping or something like that. It's really distinctive, and once you see it, you'll know that you're diagnosing beech leaf disease, because there's really nothing like it as long as you look at it the right way. It's also important to understand that we will not see new symptoms form during the growing season in those leaves. The leaves of that season only emerge symptomatic if they are only going to have the symptoms that they emerged with. That doesn't mean that there won't be further infestations. I'll talk more about the cause of the disease in a bit, but it does mean that you will not see new symptoms appear during the course of the season. Those symptoms you see at bud break are the symptoms that you'll see the rest of the season for that flush of leaves. And so the best way to really see this disease is by looking up at the leaves. Like, let's say if I'm in the forest, I'm looking up at the undersides of the leaves with light from behind. It's really, really striking, and that is the best way to diagnose that disease. If you don't see that darkening, that striping, then you're not looking at beech leaf disease. There are some other things that, looked in any other way, can be confused with beech leaf disease, but this is absolutely diagnostic. But if you look at the leaves from above, you'll see something a little bit different. It's really not easy to see the darkening, but you'll see the cupping, that hypertrophy, which just means exaggerated growth. You'll see the cupping in those interveinal bands. Between each of the lateral veins off of the midvein, you'll see extended cupping. And in fact, you'll even see, if you can look in the bottom right-hand corner of the picture to your right, you'll see that in that case, that interveinal band extends beyond the leaf margin, and we do see that in some cases as well. That's called hypertrophy. Later in the season, what you'll see is the banding thickens, it darkens, and in the case of like a hot, droughty summer like we've had in some of the past summers, that banding will drop out. It'll harden and darken and sometimes even drop out, as you can see in the lower right-hand picture here. In subsequent seasons, you have aborted bud development, and that can lead then to, if you look up into the canopy, you'll see a thinning canopy, and you'll see mortality in some cases, especially with diseased saplings, not so much mature trees, but in diseased saplings, you'll see mortality in as little as 2 to 5 years. It's not a guarantee, but that's where most of the mortality has been reported from places like Ohio, Western PA, Western New York, where they've had a few more years of experience with the disease. And so this map shows the progression of observations of the disease. We should not make the mistake of conflating that with spread. It's presumed to represent some degree of spread, but also as the disease was making its way and spreading, there was also more surveillance. So we don't want to conflate more vigilant surveillance with spread. That's an important distinction to make. But if you look at this map, you'll see Lake County, Ohio is that tiny, tiny little white spot at the southern shore of Lake Erie. And as the boxes, those are counties. As they darken to dark blue, midnight blue, we get to 2019, where you can see on the map Fairfield County and southern New York, Westchester County, Putnam County, and also Suffolk County on Long Island was where we observed it in 2019. And then I have on this map, I'm showing a red square that I've added. We don't have an updated map from the Forest Service yet, but we had reports this year. The purple squares show the result of this extended surveillance last year. I'll talk more about that in a minute, where we found it in 7 of our 8 counties. And also in 2020, and then in 2021, we have beech leaf disease now in Waldo County in Maine. That's right near where Acadia National Park is. And that's now, there's surveillance undergoing up there and in New Hampshire, and also in Vermont, to see if it's occurring anywhere else. So in 2020, based on what we found in 2019, the U.S. Forest Service funded what's called an Emerging Pest project, and that was to undergo distribution surveys so that we could hire someone to actually go out and look for the disease, basically go out scouting in forests, any place where we know beech stands are, to look for occurrence of the disease. We also put in long-term monitoring plots in Connecticut. We have 10 of them, 1 in each county and 2 in a couple of counties. Can you still hear me? Great, thanks. And so this is the result of last year's work. The red pluses indicate where we looked and found beech leaf disease, and the green X's indicate where we looked and did not find it. And we don't want to, again, make a sort of a conflation between looking for something and not finding it and concluding that it's not there. It just means we didn't find it, and I'll show you an example of how critical that is in a moment. And then the blue crosses indicate where our long-term plots are, and those we'll go back to and remeasure and observe the beech trees and see how they are faring in whether or not they are infected, because not all of these plots had beech leaf disease on them last year. So this year we'll be going back and remeasuring and making assessments of how these little plots are changing over time. Again, in 2021 now, the best way to describe beech leaf disease in 2021 is that it exploded in Connecticut and also on Long Island and even in eastern Massachusetts. We found so much beech leaf disease, had so many inquiries from people who were absolutely devastated by the beech trees in their backyards, with so much beech leaf disease, never having seen anything like it before. And we don't really know the reason for this. It's a new disease, it's a hard disease to study, easy to diagnose, hard to study. So it seems as though the distribution of beech leaf disease in Connecticut at least, and maybe eastern Massachusetts is an indication, seems to stay close to the shoreline, at least in its its heaviest manifestation. We did find, if you look up into the southern part of Litchfield County and in the western part of Litchfield County, we found a very little bit of beech leaf disease there last year, and I am getting reports from towns like Washington and Connecticut, in Litchfield County, and then in southern Windham County, down in Plainfield, right near the border with New London County. But it's really at its heaviest along the shoreline. And one thing I failed to say about beech that I want to mention is that beech is an important tree. It's an iconic tree of our forests. It makes up about 1/3 in terms of numbers of beeches occurring in our forests. And as you go north from here, and it's true for just a tiny northwestern corner of Connecticut, beeches make up one of the three important northern hardwood species of what's called the northern hardwood forest type, and that is along with sugar maple and yellow birch. And it's a very, very important tree. And in fact, as you go further north into Maine and New Hampshire and Vermont, you see much more beech co-occurring with those 3 other hardwoods in that hardwood forest type. So my favorite find this year was this picture that a resident sent me in May, and this was from Haddam, Connecticut. This is his back porch, and he's a bonsai hobbyist. And his bonsai has beech leaf disease, as you can see here in this picture, if you look at those leaves very carefully. And what's really interesting about this find is that I asked him, and he said there were absolutely no beech trees within view of his backyard or within view of his neighborhood. So this is, again, one of the mysteries about this disease that we're trying to unravel. Now, when you're out looking for beech leaf disease, and you're looking at something like you see in this picture here, it hits you. You bump into it, literally and figuratively. You can't miss it. So these are the easy finds. But when you have a situation like you're seeing in the monitor here, where you're looking up into this crown, let's say it's a little windy, the leaves are moving around, there's many layers of beech foliage, and suddenly you notice this. And you see just a few leaves in one spot with that crazy Martian striping. I hope I have that term right. I can't remember. But you see this crazy Martian striping, and even in the little blue circles, you'll see that there's tiny little bits of beech leaf disease. Now, if you happen to look up off to the right as you're doing your distribution survey, there's a trade-off. Time is limited. Do you intensively scan one little area, or do you just look really quickly and keep walking and looking around? And it's a hard trade-off, but these you could have missed. If the sun had been a little bit wrong, if there'd been more wind, you might not have seen this, and you would have characterized it with a green X, meaning that on that map, meaning that you didn't find beech leaf disease there. So these kinds of very low occurrences, this is my favorite one from last year. This is where we were putting in one of our beech leaf disease plots. And one of the things we do is we measure crown transparency. So we're looking up into the crown a lot. And we both looked at this leaf and thought, is it beech leaf disease or is it not? So we were able to reach up and grab it, barely, but we got it, brought it back to the lab, and confirmed that is beech leaf disease. And it was the only leaf on the one tree out of several that were in the vicinity that had beech leaf disease. So this is that point at which it's at such low frequency that you can easily miss it. So you don't want to conclude that beech leaf disease isn't there. You just can only say, we didn't find it. And it's not at high enough frequency or intensity that you're likely to see it. So a little bit of background now. Beech leaf gall is a name for a disease that was first observed in Japan on the Japanese species Fagus crenata in 2004. It wasn't killing trees. They didn't pay very much attention to it, but they did notice that it was associated with a leaf gall nematode, as they called it, a foliar nematode, and they named it, but only many years later in 2019. Again, not killing trees, so they didn't really pay much attention to it, but they thought it would be worthwhile to name this nematode Leidylencus crenate, and that was published in 2019. 2019 is the first year we observed beech leaf disease in Connecticut. They made the observation that they found it only on Japanese beech and not in Arboreta, where they had this leaf gall nematode occurring and the phenomenon occurring, but they didn't see either symptoms or isolate the nematode from nearby American or European beeches growing nearby. So just put that in your hat, because we're going to be coming back to that in a moment. Now, in North America, as research was being done in Ohio and Western New York in collaboration with scientists at the USDA, they identified a nematode associated with beech leaf disease in Connecticut. But they were only finding the nematode in foliage starting in around early July, not when the symptoms emerged. So that kind of put them off track. Imagine if you're a forensic scientist and you're trying to identify, what caused this. They said, well, then the nematode is probably just opportunistic. The nematode is probably just showing up and taking advantage of a tree that's already compromised. So they were looking for bacteria, fungi, phytoplasmas, abiotic causes that might be causing the crazy Martian striping. But they were able to confirm this nematode as causal to the disease by doing the kinds of experiments that you need to do that. We call it confirming Koch's postulates. I won't go into detail on that, but they were able to identify this nematode as causal. Perhaps only necessary but not sufficient, we don't know yet, but they were able to prove that this nematode is causing the disease. They injected nematodes into buds in the fall, and then they were able to reproduce the symptoms in the spring. And this nematode is very similar to, but not identical to, the nematode that causes leaf gall disease in Japan, beech leaf gall. And so we've now know this as a subspecies of Lydelenchus crenate and its subspecies McCannii. And that's based on morphology, it's based on DNA sequence data. It is truly not the same species, but they are closely related. And I just want to point out in this picture, it's also, unlike some nematodes, it is separate sexes, male and female. So you have to have both males and females, obviously, for this nematode to reproduce. They don't reproduce clonally. Another interesting observation, remember that thing I asked you to put in your hat for a little bit later, you need it now. Beech leaf disease nematodes were observed in the Holden Arboretum in Ohio, only causing disease on American, European, and Oriental beeches, and not on Japanese beeches growing nearby, nor could they isolate the nematode, the McCannii subspecies, from any of those other trees. So that seems to sort of, in a mirror sort of way, correlate with the observation in Japan. All right, so a little bit about foliar nematodes. First of all, they require water films to move, and they do move outside of the leaf. They enter and exit through stomates. And in the presence of water, juveniles and adults will enter and exit through stomates, as I mentioned. But as far as transmission of the nematode is concerned, very, very little is known yet. We really don't know. We speculate a lot of things, but we don't know for sure. Local movement, let's say within a tree or from tree to tree, is probably via rain splash. Water induces them to leave the leaves through stomates, and then you can imagine a wind comes up and a water splash sends a few water droplets containing nematodes to another leaf, and then they go into a stomate there, and now they colonize and populate that leaf as well. But intermediate and long distance movement, we know that it occurs, but we just don't know how. Perhaps on animals. I mean, they have to hitchhike on something or somebody, so it could be insects, it could be mites, it could be birds, it could be mammals. There's a scientist in West Virginia, an ornithologist, who's looking at the possibility that finches, which overwinter in forests and feed on beech buds, that if the nematode can survive passage through the bird gut, that could be a vector. And it's not one or the other. It could be any or all of these things. Scientists at the USDA isolated arthropods of various types like spider mites from foliage on beech trees in the Holden Arboretum, brought them back to the lab, did electron microscopy, and showed that there were nematodes on the legs and the backs and just different parts of these spider mites. So it is very possible that spider mites, which can move on wind currents long distances, could pick up a nematode or two on their body and then take it to a new tree. And also, we don't yet know about nurseries, whether or not European beeches, which are an important part, maybe small but important part of the nursery industry, whether or not European beeches might also be a way that the nematode is getting around. So in 2019, once we found beech leaf disease on Long Island in New York, we joined up with other scientists and formed what we call a beech leaf disease working group, and it includes people from these various organizations, including the USDA, both the research division and the Forest Service. And we've learned a little bit more about the life cycle of the nematode. For one thing, we know that in the wintertime, the nematode does a very good job of sequestering and remaining viable in buds. So in the fall, they start moving from leaves and going into buds, and they are very attracted to buds, and that's been demonstrated in the lab. In spring, by bud break, we don't find nematodes any longer in the buds, and we don't even find them in the symptomatic leaves. Those symptomatic portions of the leaves we don't isolate nematodes from those leaves. They might be there and we just can't get them out. It's not that easy to do. But I developed a molecular marker for the nematode. It's highly specific to this subspecies. And what we can demonstrate is that the nematode is present in whatever form. We suppose eggs, or it might be that there are just recalcitrant juveniles and/or adults that just don't feel like leaving the leaf at that point. So for an example here, distribution in early season symptomatic leaves, picture this leaf, imagine taking a hole punch and chopping out, or hole punching out pieces of symptomatic tissue, and then if we apply this molecular marker to each of those individually, the bands show where we can prove presence of the nematode, and the absence of the bands indicate where we don't find the nematode. So it suggests that in these early symptoms leaves, the distribution of the nematode is very sparse. And that could be eggs, and it could be just a very, very few nematodes that have survived over winter, or it could be both. In July through October, we do start finding nematodes in leaves, like now. Now we can take leaves into the lab, we can float pieces of leaves in water, and easily isolate nematodes out of those leaves. And what's interesting is the population densities inside those leaves increases dramatically, exponentially, in fact. This is a research paper from Sharon Reed in Ontario, where they studied year after year. I forgot to mention that Ontario started seeing beech leaf disease a year after it was seen in Ohio, and they have it quite widespread now. And actually, if you look at this graph, September and October, the numbers are even greater. I needed a lot of nematodes to do a whole genome sequence. I was able, with leaves isolated in September, to get nearly 500,000 nematodes from a single leaf. So that's how much the populations increase. This is some really beautiful electron microscopy that was done at the USDA by Gary Bauchan, who I have to say sadly passed away due to COVID earlier this past winter. And I'm going to show you the healthy cross-section of a leaf from a healthy leaf and a cross-section of a leaf that's infected. And these leaves are in cross-section, so the underside of the leaf is the bottom part and the upper side is the top part. So if you look in this picture here, you can see the nice organized architecture of a leaf in cross-section. The palisade layer on the upper part, and the lower part is the spongy mesophyll. And it's very structured and organized as a leaf needs to be. And this is an infected leaf. You can barely make out the palisade layer, or at least just remnants of it, the detritus of all of that feeding of the nematodes. And you can actually see the castings of some nematodes. They're the sort of bigger, longer, noodly-like things in the lower part of the spongy mesophyll, which is also heavily disintegrated after having been fed on by the nematode. So what about control? I have to, wrap it up here at some point. There's lots more I could talk about, but I'm limiting myself to really just giving you an introduction to this disease. There's research that's been underway for several years now, even before the nematode was proven in 2019 and then published in 2020 to be the cause, using various kinds of nematocidal compounds like emamectin benzoate, which is the most water-soluble of the ivermectin group. And this is through trunk injections, and there's a lot of reluctance to use trunk injections. You're dealing with a thin bark tree, and down at the soil line, injecting the macro injections. Micro injections are a more reasonable approach, and that's also been tried. But there's really not been seen much efficacy with this method. As expensive as and invasive as it is, it doesn't seem to have much efficacy. On the other hand, many of you who are in land care, or at least work on trees, know about agrophos, or polyphosphites. And that now, especially this year, as of an email I got only 2 weeks ago from Ohio, that saplings that have been treated with soil applications, whether soil injections or soil drenches of polyphosphite, that this year, trees that were looking like they were hanging on a little bit better than control trees, this year trees have really recovered dramatically. And that's even the saplings, which really take the hit much worse than the mature trees. I haven't even had a chance to talk about beech bark disease. Many of you know about beech bark disease, but the dynamics of beech bark disease in mature trees relative to saplings is kind of the opposite of beech leaf disease in a way. And with these kinds of soil applications, they're seeing very promising results. And so this is anecdotal. It's not been published. They're still collecting data, but it is something for people to try. It's much less expensive and certainly not invasive the way that injections are. Now, my research, I'm really focusing on identifying pathways of how the nematode is getting around. And since we can't, put the little tags like you put on bird legs to study migration of birds, we have to use molecular markers. So we did that in order to use those markers, to develop those markers that we used for early detection of the nematode, and also to allow us to discriminate between the two subspecies. We would like to know if the Japanese subspecies is here, but more importantly, if it ever comes to it, this would be an ideal marker to take to Southeast Asia, where it may be this nematode came from, if there's ever an interest in determining where did this nematode come from. So I want to use this for studying pathways of spread and using a population genetics approach, which is my area of research. For that, we used a whole genome sequence of the nematode and developed DNA fingerprinting markers. And if you've ever watched any crime scene TV shows, that's what they use to determine if someone is the father or if someone is the murderer, et cetera. In crime scene forensics, this class of DNA fingerprinting markers are used broadly in various kinds of ecological studies, and a very powerful tool. And that's what we are right now in this, almost at the finishing stages of developing and pretty much ready to deploy, so we can start asking questions about where, for example, the Maine outbreak, where did that come from? Did it come directly from Ohio? Did it come from Connecticut? It would be helpful in allowing us to narrow down how this disease is spreading from site to site. If you need more information, we have an updated fact sheet, and I think there's plenty of copies in the publications tent. It's also available online at our website. If you're concerned that your tree may have beech leaf disease, you can send photos to Dr. Lee in the PDIO, me if you have to, but it's pretty diagnostic, or contact your arborist or your local tree warden if you see the disease and are concerned. Many people are reporting it because, as I say, once it's really, really prevalent on a tree, you can't miss it. So in summary, beech leaf disease seems to be spreading rapidly in the Northeast. It's caused by a newly described nematode named Leidylenchus crenate subspecies McCannii. The symptoms in the current year are due to nematode infestations into buds the previous year. So when people say this is the first year I've had it, I try to remind them, no, you had it last year. You had to have had this disease here last year. And these polyphosphite or agrophos type soil applications appear to have a salutary effect on the disease. But more research, and therefore more research funding, is needed so we can proceed in learning more about this disease, and also learning through forest ecological studies what is going to be the long-term impact on forest composition. So I'd like to acknowledge these folks. I'm not going to name them all by one at a time, but these are all people who have been important, people in divisions who have been really important in helping us move forward in learning about this disease. And with that, I think I have a little bit of time for questions. Yes, sir.
Is the polyphosphite insecticide that you mentioned available and commercially available and registered in Connecticut?
Well, because it's considered a fertilizer, it doesn't really need to be registered for this type of use.
And what kind of a fertilizer is that?
Well, it's called, one of the commercial brands is Agrophos.
Agrophos.
I can't tell. She'll come around with the microphone in just a moment.
Does this have any implications for how we collect leaves in the fall and process them? It sounds like the nematodes stay in the bud, but what about the fallen leaves?
The fallen leaves in that study from Ontario, they were not able to identify much other than a few viable nematodes, but not by the end of winter. So they really do depend on that sequestration in the bud for winter survival.
As far as applying the agrifos, is it a soil injection or a drench?
I think they've tried both, but I think that the main applications were soil injections.
Thank you. Do we know if people are capable of spreading this around if you're in a forest situation where there's a lot of beech leaf disease, or no?
Well, we don't know, but one could speculate. I mean, we should always be cautious when we go out in the field. We're spraying the undersides of our shoes with ethanol when we leave a site. Ethanol is if there's a viable nematode, it's not going to be viable for very long in ethanol. They turn from little wiggles into these straight little pin-like structures. But one thing to keep in mind is that until July, we can't get nematodes out of leaves. But I suppose if someone picked up a bunch of leaves and put them on the forest floor, the only thing those nematodes in those leaves have to do. And also, one thing that's really interesting is that leaves that we collect in the spring that we don't isolate nematodes from, if we put them in the fridge or leave them out on the bench, whatever process is necessary for that leaf to suddenly in July start revealing or releasing nematodes, that process is cut short by removing the leaves from the tree. So I don't know that leaves moving around are really an important source. I think it's reasonable to speculate, knowing what we know about the biology of the nematode, that it's the rain splashes that result in local movement and within tree and among tree spread of the nematode. Yes, sir.
What's the long-term prognosis for an infected tree?
For mature trees, it seems as though they hang on. But we only have 9 years of data. So, and 9 years is just a wink of an eye for a tree, so we don't really know. I mean, until we've had more time with this disease, this tree is naive to the nematode. You know, the idea that trees will evolve resistance, whether or not there is resistance, there's some thought that when we see a tree that has beech leaf disease next to another tree that doesn't, perhaps that's resistance, or perhaps that's just the result of the random way in which this nematode seems to be able to get around. We just don't know. That's going to require years of study to be able to make conclusive statements about that.
Bob, I have, in my mind, kind of a time disconnect. The photo that you have up now is early season, and we're seeing that interruption of the translucence. The photo that you showed from the USDA, from the electron microscope, and all the damage inside the leaf, the physical damage, is that later in the season, or is that what is causing this early season symptom?
That's later in the season.
So what's causing the early season symptom?
We don't know for sure. Okay. Probably some kind of enzymatic, it might be strictly a response of the leaf, because that's damage that's caused in the little leaf primordia inside the bud. And physiologically, those leaf primordia are very, very different than a mature leaf, which is probably why late in the season we can extract nematodes out of adjacent bands that are not symptomatic, but nematodes have colonized. And they can colonize asymptomatic bands or asymptomatic leaves. They can colonize those, they can reproduce in those, but you're not going to see symptoms as a result of that infestation. So those symptoms are unique to the leaves that emerge from the bud.
So it's going on in the bud?
It's going on in the bud, yeah.
Thank you.
I think one more question, and then we have to move on.
How do those bands look in the dark purple-leafed European cultivars?
Yeah, that's a good question. And actually, the two samples that I got last year from Lexington, Massachusetts, and Plymouth, Massachusetts, were both copper beeches. In fact, we don't have any American beeches yet from Massachusetts. The main trees were American beech. But you can see it if you're looking, again, you need to look at the underside of the leaf with light from behind. You put the leaf down and look down at it, you will think no BLD there, but hold it up to the light and you will see beech leaf disease, and you can see it in copper beech. The beech tree that I'm curious about is fern leaf. The European fernleaf beech, that cultivar, I don't know what the symptoms would look in that because it's so highly lobed. So maybe there are fernleaf beeches out there with beech leaf disease and you're not seeing it. I just don't know. But bonsais can get it, so we know that. I think that's the last question.
Okay, thank you. So next we have presentations of 2 awards. The first is the Outstanding Young Farmer. This is an award that's actually given each year, and it's usually announced at the Ag Day in the Capitol, which is April of each year. But this year the event was virtual, so in the hopes of better recognizing the grower, we wanted to have the event here as well, but unfortunately she's on vacation. But there is a proclamation from the governor, which I will read. So the 2021 Connecticut Outstanding Young Farmer Award goes to Suzie Flores of Stonington Kelp Company in Stonington. So Suzie is a former market development executive who owns and operates Stonington Kelp with her husband Jay and 3 children. The vision for the farm began 5 years ago in 2016. And the following year was up and running through a partnership with Greenwave, a nonprofit dedicated to supporting the next generation of ocean farmers. Today, the farm is one of the largest commercial seaweed farms in the state, selling food-grade sugar kelp to local restaurants and shops. Sugar kelp is the only variety of seaweed that can legally be grown in Connecticut, and it has a slightly sweeter flavor. While the nutritional benefits of sugar kelp are well known, the positive role this native sea vegetable can have on the environment is what really drives the Flores family. Her farming methods highlight the importance of regenerative and sustainable farming for the ocean. Sugar kelp absorbs carbon and nitrogen from the water while it grows, directly addressing climate change and mitigating the impacts of ocean acidification. And this is signed from Governor Lednemont, Executive Chamber. So our department head in the Valley Laboratory is going to accept this award on Suzie's behalf. There we go. Thanks, Jason. And the second award is also another annual award. It's the 2021 Connecticut Century Farm Award. The Century Farm Award is given each year by the Connecticut Agricultural Information Council to a farm that's been in continuous operation for over 100 years. And this year, the 2021 Connecticut Century Farm Award goes to March Farm in Bethlehem, Connecticut. They were hoping to join us today, but at the last minute could not. So again, here's a proclamation signed by Governor Lamont. March Farm is a 4th-generation family farm located in Bethlehem among the beautiful rolling hills of Litchfield County. Purchased in 1915 by Thomas and Rose Marchakadis, the farm consisted of 114 acres and supported 15 cows and 2 horses. In 1937, Thomas and Rose's son Matthew and his wife Anastasia bought the land and the stock. At that time, a diversified farm was started and operated as such for many years. The first tractor was purchased in 1939. During the 1940s, poultry and dairy barns were added to accommodate 50 cows and approximately 600 chickens. During the 1950s, 14 acres of adjoining land was purchased and apple orchards were planted. Throughout the next 3 decades, stock and fruit production were increased until there were 100 dairy cattle, 40 acres of fruit trees, 5 acres of blueberries, and many acres of sweet corn, cabbage, potatoes, squash, pumpkins, other vegetables, and hay. On July 1st, 1977, the farm was purchased from Matt and Anastasia by their son Thomas and his wife Susan. In 1988, some changes were made. Thomas decided to sell the cows and concentrate more on producing vegetables and fruit. A commercial kitchen was also installed in order to accommodate a bakery business to supplement produce and other items sold at the farm store. The next year, a greenhouse was added to produce quality tomatoes. Since that time, 11 more greenhouses have been added to produce a variety of tomato types, cucumber, squash, lettuce, and salad greens, in addition to many outdoor vegetable fields. Orchard sizes have increased, and as has the production of sweet corn. During the 1990s, Pick Your Own became increasingly popular and has been encouraged for berries, fruit, and pumpkins. So again, this proclamation signed by the governor, 2020 Connecticut Century Farm Award to March Farms. Thank you. So in addition to the proclamation, there's also a plaque, which is very heavy, which goes to March Farm as well. And we're going to keep that right here. Thanks. Okay, so next up, I'd actually like to introduce Terri Jones, who's Vice President of our Board of Control. And I see him in the back, but he doesn't know I'm talking to him, so maybe somebody could run and grab Terri. Here he comes. So in addition to being as the Vice President of the Experimentation Board of Control, Terri's also on the board of the Community Foundation of Greater New Haven.
I'm chuckling because I was here 20 minutes ago and the place was full. Didn't know I had that effect on people. So I don't even think my family is here. Well, so I'm speaking as my role as Vice President of the Board of Control. As you may or may not know, the governor is president, and both he and Dan Malloy and Jody Rell, we've had a run of very supportive governors. But first, I wanted to thank the station leadership, Jason Wade and Mike Last, and the entire staff, who I can assure you this past year have been working their hearts and minds out on behalf of the success of the station. And I will never forget, a year ago, there were just 3 of us here, because it was all done virtually. And that was quite an eerie experience. The Board of Control, I also have enormous appreciation for. As you may know, they are comprised of a representative from Yale, Wesleyan, UConn, Commissioner of Agriculture, and the governor, and a couple of the governor's appointees. So it's a great collaborative, a lot of wisdom. in that, on that board. So I've been attending Plant Science Day since I was 6 years old. Not every year, but many years. And that was almost 70 years ago. It's kind of scary. And I've known 6 of the directors. And how many directors have there been? I think 11 altogether since 1875. And I always appreciated that my parents would bring me as a child. And I appreciate now that our son and daughter-in-law, who are here or en route here, and my wife are all collaborative on the farm, and it allows me time to be involved with station affairs. Connecticut Agricultural Experiment Station is not only the first in America, the original, but it's also, with the possible exception of Ohio and Columbus, the only one in a major city where our campus is. So we are unique. And I have had the joy the last 2.5 years serving on the, also on the board of the Community Foundation for Greater New Haven. And I'm very excited that I think we're going to engage in some more urban agriculture projects, and the opportunities are enormous. And I think our speaker, the Johnson speaker, lecturer, will have something to add to that. But I did want to, 2 of my colleagues are here, and I just have Carlton Highsmith and Carlton grew up on a farm and in eastern North Carolina, and went on to have a major manufacturing business in packaging, and it was the largest in Connecticut, owned by a minority. And Valerie Schultz-Wilson is also on the board, and believe it or not, she's been on the board for 1.5 years, and this morning was the first time we met face to face because everything's been virtual. And she is extremely involved in community affairs and justice, and single-handedly, in my opinion, turned around the Urban League of Southern Connecticut, which was kind of languishing, and now it's in 4 counties. So these, and that's just the tip of the iceberg of the rest of the New Haven board, like our board of control. It's amazing people. And our CEO, Will Ginsburg, will hopefully be here as well for Brent's lecture. So lastly, I want to announce, and I don't want to steal the thunder, but come on, the Lieutenant Governor is here.
Okay. Thank you.
How are you?
Terry, nice to see you.
Yeah. So this is news to you, but it's near and dear to your heart.
Okay.
Because for a number of years, we had a program with the Women and Girls Fund at the New Haven Foundation. And Sharon Douglas, one of our outstanding women scientists, and others, Wade Elmer worked with them, mentoring at the station. and getting these girls from the New Haven area in from the high schools. And Dr. White has reached out, and there is interest in several of the scientists to starting that program again. So I am really pumped up about this, because what better way for a young high school girl who might not have the opportunity to be brought in and be mentored and be at the ag station in a laboratory. That's the way to really learn. And we've even figured out we'll probably, because transportation has always been an issue, we have Uber now. So
Yes, we do.
we'll secure funding. We're going to get these girls in.
I love it. Love it. All right.
Initiative number 2. We have just hired.
List. Okay.
Yeah. Well, I know that one's really special to you.
It is.
We have hired our first urban agricultural scientist.
That's pretty cool.
And food security person.
Excellent.
Lee is going to be joining us in October. And Carlton Highsmith.
Oh, hey, Carlton. Love the hat.
Well, that's his farm hat. He's working with Eric Clemens with Concord on the 7.5-acre Dixwell Plaza community-driven reincarnation and redo of it. And guess what they're going to have on the roofs of the parking garage? Because they're going to have several restaurants.
Okay.
They're going to grow their own food.
That sounds
So there'll be rooftop gardens and rooftop greenhouses.
They're doing
And
that in New York
our Yukon is having some input. In fact, they were on the search committee with this scientist, and I believe there's a Yukon extension person going to be involved with urban agriculture.
Excellent.
So that's pretty cool. And then Will Ginsberg, who I don't think is here yet.
Haven Foundation, I know him.
But he is working with the new owner of some farmland in New Haven, and we're going to be working on opportunities for residents to do some farming there. So all in all, what it boils down to, I think I might have invented this phrase yesterday thinking about this, the poetry of collaboration, where we've got 2 great institutions. We're in New Haven, and we're going to ramp up working together on behalf of New Haven and things related to food security, and agriculture.
Awesome.
So with that, I'm through. And I think...
Is it my turn?
I think it might be your turn.
Okay.
Is that right, Jason?
Okay.
So...
Thank you. A round of applause for Terri Jones, who is my unofficial agricultural and farming senior advisor, not just to me, but also to Governor Lamont. And I love him keeping me informed. And pre-pandemic, he actually delivered a box of the most delicious strawberries to my office and to the governor's office. So Terri, thank you for everything you do for our beautiful state to promote agriculture and for the experiment station. And good morning to all of you. I'm so pleased to join you for the 111th Plant Science Day, and I gotta say it's much nicer to be here in person rather than virtually like I was last year. I want to thank Dr. Jason White for his leadership and for the invitation today. This event is a wonderful opportunity to learn about scientific research in agriculture and food safety, the environment, forestry, and public health. The Connecticut Agricultural Experiment Station has been historic in our country because it was the first agricultural experiment station in the United States, and it's been since 1875 at the forefront of addressing the challenges that our residents face every day. It's been a leader and made significant contributions right here in our state and across the country. You should know that I am a farm girl by background. I grew up on a potato farm in Middletown, and it is still there. And yesterday, the city of Middletown voted to preserve the 40 acres of farmland that has been in our family for 100 years, to preserve it for open space and for continued farming activities. It's going to be called the Bysiewicz Farm in honor of not me, but my grandparents, my dad, my uncles and aunts that all worked on that beautiful farm. And what's great is, like this one, it'll be preserved for many, many generations to come. I want to thank the folks at the Agricultural Experiment Station for all the work they're doing, particularly right now, tracking West Nile, triple E viruses, the education they're doing about the spotted lanternfly, emerald ash borer, and the box tree moth. This information is really important to the people of our state, and we appreciate everything that you're doing to keep us safe. And we know that there's potential for the spotted lanternfly to have a significant impact on our orchards and our vineyards here in Connecticut, and know that for every spotted lanternfly female that is killed, there will be 90 fewer spotted lanternflies next year. So help us join that effort. And I just want to say thank you for what you do for agritourism in our state. You'll be hearing from our great ag commissioner, Brian Hurlburt. Agriculture means $4 billion to our economy here in Connecticut, and more than 20,000 jobs. We are home to some dozens of beautiful wineries, and now more than 100 craft breweries. And a lot of them actually grow their own hops. And I like to say we have 110 craft breweries, with many more in the hopper, because every town is now seeing multiple craft breweries spring up, and people are coming from all over our state and our country to visit them. So we know that lots of great things are happening in our beautiful state, and a lot of them are because of the hard work that the Agricultural Experiment Station does every day. So I want to thank all of the great folks who are part of the team, and hope you have a very enjoyable and educational day. Thanks so much. It's such an honor for the governor and I to represent you every day.
Well, thank you to the Lieutenant Governor for coming. That was quite an honor. So our next speaker is actually going to be President of the Experiment Station Associates, and Ms. Cheryl Cappiali, and she'll be making a few comments and then discussing an award that she gave to one of our scientists.
Hello, everyone. What a pleasure it is to be here in person today. We're very fortunate to live in a state who values science in the forefront, and nowhere are innovative ideas more successfully tested and proven than at our experiment station. We, the Experiment Station Associates, are dedicated friends group whose mission is to support the scientists and their research. I encourage you to stop by Barn A and register with us for a new membership or renew your membership. This year, we gave an Early Career Scientist Award to Susanna Carriel, and she's here with her lovely research assistants. And the research assistant is this year Jackie Lemon, and she also has another assistant that she was so lucky to acquire through another grant. So, Susanna, thank you.
Thank you, Cheryl, for the introduction. Lieutenant Governor, honorary guests, and dear station staff. I am truly honored to be the recipient of the Early Career Award this year. This grant actually marks a very important milestone on my scientific career, as it is the first research grant that I won as a principal investigator. Yay! So completing this project would not have been possible without the support of the Experiment Station Associates. So thank you, ESA, very much for supporting my research. The project that my collaborators and I started this year relates to drought stress responses in trees. One particular interest of mine is how we can grow healthier urban trees. So you may ask why urban tree health is so important. It's because of the connection of tree health to public health. So basically this means trees make us happier and they make us healthier. And if we have a lot of healthy urban trees, we will also be healthy. The research shows that just by looking at trees, you can get health benefits such as stress reduction, which helps us to live nice, long, happy lives. Why tree health is such a hot topic is because of the increasing tree stress globally related to the global climate change. Too much stress can eventually cause the trees to die, which is not good for anyone. Not for the trees especially, but it's not good for us either. One culprit involved in tree mortality is drought. Severe droughts have become more common, which has increased tree mortality in forests, and especially in urban areas where the stress levels for trees are already pretty high. Therefore, new management approaches to improve tree stress tolerance and survival in urban and agricultural settings is desperately needed. The project that my collaborators and I aim to address this problem of increasing tree stress. The project that we started this year tested if nanoparticles could be used to manage drought stress in trees. Previous studies here in the experiment station and elsewhere indicate that various nanoparticle compounds can benefit plant health and improve drought tolerance in agricultural crops. Our goal was to test this concept on trees, one of the first studies conducted on nanoparticle research in the whole world. The preliminary results from the project indicate that copper oxide nanoparticles may have beneficial impacts on tree growth and water content, which might help tree seedlings to survive abiotic stress. In case you're interested in hearing more about the project, please visit our exhibit in plot 30. I also want to thank my amazing collaborators from three other departments in the Connecticut Ag Experiment Station. Dr. Nubia Suversa-Mena from Analytical Chemistry, Dr. Washington da Silva from Plant Pathology and Ecology, and Dr. Blair Stephen from Environmental Sciences. Last, I want to present you to the dynamic duo who made this project happen. So Jackie Lemon and Aiden Florio. Without their hard work and persistence, the chestnut seedlings would have long gone and dead, and we would have no results to present. So please welcome Jackie and Aiden.
You want me to go first? Hello. Hello, I'm Aiden. I've worked with Susanna for the last couple of months, working with the chestnut trees. I've had to incredible time here working at the experiment station. Not my first job, but actually my third. Could go on to say that the station is a fantastic place to work if you're trying to do some scientific experiment work, work with any of the departments, or if you're just a citizen science, or just a average person like everyone here who needs something done, whether it's with ticks, chestnut trees, mosquitoes. Station is a great place to be, lots of information, lots of knowledge, and we're always here to help you and give you information that you need.
Good morning, I'm Jackie. This is my first experience at this station. I've had a lot of good training that I haven't had before, which is going to prepare me for grad school next year, so I'm really excited about that. And hopefully I'll continue my research in the future on tree stress, and yeah, it's been good. Thank you.
So just want to close with a thank you to everyone and hope you enjoyed the 111th Plant Science Day.
Okay, thanks, Susanna, Jackie, and Aiden. So next up, I have the pleasure of introducing our Samuel Johnson Memorial Lecture. Our lecturer today is Brent Peterkin. He's the Executive Director of Gather New Haven, which formed on January 1st, 2020 as a merger of New Haven Farms and New Haven Land Trust. Brent's a native of New York City, and he comes to Gather New Haven after almost 6 years as the statewide director for Project Longevity, which is a nationally recognized program of the U.S. Attorney for Connecticut that's focused on reducing gun violence in Hartford, Bridgeport, and New Haven. Brent has also served on the Governor's Youth and Urban Violence Commission and the Bridgeport Mayor's Initiative for Reentry Affairs. He is a board member of Emerge and CT Against Gun Violence. Previously, Brent was also a Zoom Policy Fellow in the Office of the Governor, working in support of the Office of Early Childhood, focusing on parent engagement and fatherhood development. Brent has also worked with the Yale University-based Global Anti-Poverty Initiative and supported at-risk high school students with emotional, behavioral, and learning disabilities in Yonkers, New York. Brent studied public policy and public administration, nonprofit leadership, and social impact strategy at Fordham University, Northwestern University, Howard University, and the University of Pennsylvania. His commitment to social change has led to recognition as an honoree of Connecticut Magazine's 40 Under 40 class of 2017 and 100 Men of Color 2017 honoree. So we're honored to have Brent here with us today to give the Samuel Johnson Memorial Lecture, and we'll load his presentation up right now.
Podium mic is on. Podium mic is on? All right, we'll use the podium mic. That works. Well, it's still morning, so good morning, everyone. All right, it's a pleasure to see you all today. Very appreciative of this opportunity. As Jason shared, my name is Brent Peterkin. I'm the Executive Director of Gather New Haven. I'm honored to be the Samuel Johnson Memorial Lecturer today. I prepared a short slide deck. Just wanted to share a few stories with you all, talk a little bit about cities and urban ecology or urban ecosystems, and some of the challenges and opportunities that lie ahead of us. So once again, I appreciate this opportunity, and let's go along for a ride. So as you've probably read in the promotional flyer and the invitation for this event, I'm going to be talking about urban ecosystems and cultivating communities. And I know I'm preaching to the choir here, but I'm just going to share a few definitions. You can all see it on your screen, but essentially when I speak of ecosystems, I'm talking about the entirety of interactions. When I speak of urban ecology and human ecology, I'm speaking specifically about the interactions between people and their environment. These are constructed environments with elements of the natural world, as pretty much no matter what we do, we cannot keep nature out. And in fact, we should invite it in whenever we can. And this subject matter is meaningful to me because of where I grew up from. And as Jason shared earlier, I'm originally from New York City. And my experiences growing up in a large city inculcated a sense of appreciation and essentially I came to believe that nature can heal and grow communities, and communities can restore and sustain nature. I grew up in New York City during the 80s and the 90s. It was a volatile period in the city. Like most urban cities or urban environments, you're surrounded by concrete, steel, and glass and bricks. Although in my neighborhood, we did have some parks, we did have some green spaces, but we weren't offered an opportunity to have an immersive experience. So we did have some amenities, but again, in the surrounding neighborhoods and in my neighborhoods, those amenities were few and far between. The time in which I grew up in New York City, the black and brown male gun violence and incarceration rates hit historic highs in that city. And so although I grew up in a very stable household with loving parents, like any environment, man-made or natural, once you're in the environment, you're subjected to the circumstances. And so whether you were a kid who were on the wrong side of the tracks or you were on a positive trajectory, again, you can become a victim to those circumstances that existed outside of your doors. When I was 13 years old, I had a life-altering experience, actually two of them. I had just finished middle school, and the summer before I entered high school, I went to a house party, which I should not have went to. My parents did not know I was going to this house party. Don't tell them. But I went anyway with my friend Danny. And the house party was on the 7th floor in an apartment. And some guys came to rob the people who were in the house party. And some of the guys in the house party had firearms and drew their weapons. So there was a standoff. My friend Danny and I actually spoke to each other as we ran to the back to the bedroom and said, should we hide under the bed or try to climb out of the window? I was 13 years old. I realized then, and based upon prior experiences, that my mortality was possibly in jeopardy. Not because I was involved with the wrong activity, but again, once you're outside of your home, you're subjected to the circumstances of your environment, man-made or natural. Later on in that summer, after playing a pickup game of basketball, a friend of mine got into an argument with some other youths. One of the youths pulled out a firearm and started firing shots aimed in my direction. I froze, came to my wits, and took off running. I normalized that reality. I came right back to the same basketball court. In fact, I didn't tell my father about that incident until I was about 30 years old. What I didn't realize at the time was that I was internalizing this trauma. And I was also trying to navigate the perils of the urban environment. And these are things that individuals who are not emotionally developed and are afflicted with trauma can easily succumb to. But fortunately for me, I was offered an opportunity by a school guidance counselor and a little bit of my ambition to find a summer job. See, I had started work when I was 12 years old. And the reason why I started work when I was 12 years old, so I wanted a pair of Jordans. My father challenged me and said he's not paying for those sneakers. And some of you are old enough to remember a brand of shoes called Ponies. So my father picked up a pair of Ponies and said, you can get these for school. And I said, I can't wear those to school. He said, if you want to get the sneakers that you want, get a job. So that set me off, starting with my first job at the age of 12. When I was 14, school guidance counselor shared an opportunity for me to work at a place in the Riverdale section of the Bronx called Wave Hill. Now, if you've ever been to Wave Hill, it's a beautiful environment. And I was 13 years old, heading into this environment for the first time, freshman in high school, and I had never seen anything like it. I was immediately enamored with the environment that surrounded me. We had to be there at 9 a.m. We spent our days doing ecological restoration work, which is new vocabulary for me, but we were literally in the weeds. We were clearing out invasive species. I come to like the environment so much that I started to show up to work early. In fact, with the 9 a.m. check-in, I would get there around 7:30. And what I would do was I would walk down the trails into the woodland areas by myself, about a half a mile in. I've never done anything like that before. But I would sit on a log, or just stand around and take it all in. And I felt something that I never felt before. I felt safe. I wasn't white or black. I wasn't in jeopardy or in fear. I felt completely at home. It was almost as if the environment was offering me a welcome. That experience left an indelible mark in my mind. And that experience is what led me to Gather New Haven. Once I had the opportunity to work for an organization that focused on environmental justice and food justice, I took a leap of faith because I felt like I owed nature something in return. I needed to reciprocate what I was given many years ago. And Gather New Haven is a nonprofit organization, and as its namesake implies, we focus on urban agriculture, environmental education, youth development, and also property management. We manage 6 nature preserves in the city of New Haven, and a whole bunch of other things that I'll get to in a few seconds. But Gather New Haven, as Jason shared earlier, consists of 2 preceding organizations that were formed by way of a merger, which is quite rare in the nonprofit sector. But there was an opportunity for 2 organizations to create symbiosis and synergy and offer more meaningful impact for environmental issues and also for social impact issues. Now, I challenge you all, well, hopefully you won't have to experience it, but this is a forewarning in case history repeats itself. Do not start a new job during a global pandemic with subsequent to a merger. It is quite daunting, and there is no work-life balance at all. So as I shared earlier, Gather New Haven manages programs and properties. The programs that we offer are a farm-based wellness program where we focus on chronic diet-related illnesses. We work with people in low-income communities or from low-income households who either been diagnosed with a diet-related illness like diabetes, hypertension, overweight, obesity, or at risk. And essentially the goal there is to spur cognitive behavioral change, to change their attitudes towards food and the social dynamics connected with eating so that they will be in a position to mitigate or minimize their risk of developing a diet-related illness or be able to better manage their disease. We also manage an initiative called Growing Entrepreneurs, which focuses on high school-age youth who are enrolled in New Haven public schools. And these youths are employees of Gather New Haven. They work throughout the year. Their hours increase during the summer months, thankfully to the Youth at Work program offered through the City of New Haven and with the support of donors. These youths are also offered an opportunity to start their own business venture. And it just merely has to be connected to environmental or food-related matters. But essentially, it's their own adventure. They choose what they want to do, and they're able to pressure test whether or not their ideas are viable. We also manage 57 community gardens with a very small staff. So we rely a lot on volunteer support, and we rely a lot on community-led initiatives and community leaders, throughout the city to take ownership of these green spaces, and we provide the material resources and, when necessary, technical assistance. And then lastly, we have our Schooner Youth Camp, which is an environmental education and learn to sail program that operates out of the Long Wharf Nature Preserves. Speaking of the Long Wharf Nature Preserves, it's among 6 of the nature preserves that Gather New Haven manages. As I said earlier, we manage 57 community gardens, 7 urban farm properties, and we operate a farm stand. We were operating 2 farm stands last year. We now operate 1 farm stand at our Ferry Street Farm location. It's quite daunting, but that is the work that we do in the city of New Haven. Speaking of the city of New Haven, as many of us experienced last year and for the first half of this year, the city of New Haven has gone through significant challenges. Gun violence has reverted back to historic highs. We know that food insecurity and unemployment have been exacerbated as a result of the pandemic. We know that our youth are struggling with learning from home to remote learning or through a hybrid schedule, and they're also losing opportunities for structured out-of-school time. Thankfully, the federal government, the state of Connecticut, and the city of New Haven provided resources for summer enrichment and expansion opportunities, which supported organizations like Gather New Haven and others to be able to offer more opportunities to our youth. Now, with this confluence of issues, it's easy to just throw your hands up and say, well, what can we do? We just have to wait it out. But I asked, and what many people have asked, including the healthcare community, is what can the natural environment do to help? That is our immediate resource. It is reachable and accessible to us all, and it provides a plethora of solutions. What are some of the solutions that New Haven has to offer? Well, we have major parks, we have walking trails, we have public playgrounds, community gardens, watersheds, and coastlines. We also have an immense array, and some argue that New Haven has too many nonprofit organizations, but that's our strength, not our weakness, and I stand on that. We also have a community of environmentalists and educators. And here we have an opportunity to focus on human ecology, how the natural environment can positively impact the residents that live in an urban setting. How can the natural environment offer a resource to some of the challenges that our society and our communities are facing? Well, a couple of things. And it seems kind of trivial, but you'd be surprised how much we don't get out of the house, even pre-pandemic. Many of us are inundated with work, trying to struggle between the responsibilities at home and our professions and other activities, and we don't find enough time to take it all in. I found that many researchers agree that what I experienced many years ago is exactly what's needed now. We need to encourage our residents to take advantage of the amenities that are in the city of New Haven and in surrounding township. We also know that the ecosystem provides what some researchers call services, the benefits of the ecosystem, which include mental health reduction, stress. There's research that points towards community gardening environments, natural environments like woodland environments and other environments that have offered therapeutic benefits that have been supportive of women and men who've been in domestic violence or intimate partner violence situations. We also find that it also facilitates therapeutic benefits for our veterans who are experiencing post-traumatic stress disorder. Also, community gardens, just growing something, right? Getting your hands in the dirt, feeling the texture of plants and roots, kinesthetic learning can help with youth who are on the spectrum. That's our remedy right there. We know that green spaces also help communities address public health issues. So throughout the city of New Haven, there are people who are growing in community gardens, there are people who are growing at home, and a lot of that has been spurred by the pandemic. So people are seeking to eat healthier, to eat better. Now that matters in communities that don't have grocery stores that offer fresh and organic produce. These are neighborhoods or households that are food insecure. Another benefit that comes out of that is immunological resilience. Sunlight helps to strengthen cellular membranes, vitamin D. Eating green leafy vegetables helps to strengthen cell membranes, vitamin A. That makes you more resistant to viral infection. Nature, once again, provides a remedy for us all. We need to encourage this, we need to promote it. Obviously, the economic circumstances need to be in place to facilitate more growth in this area. We also know that having more green spaces in the urban environment helps to reduce or mitigate the impact of heat islands. In certain neighborhoods, you can experience as much as a 10 to 15 degree difference in temperature than in other parts of the city that have green spaces. Oftentimes, those areas that are called heat islands are in low-income, Black and brown neighborhoods. One of the challenges that comes along with greening spaces and investing in green space infrastructure is ecological gentrification. I spoke to a community garden recently, and she leaned into me and she says, Brent, you know what? they never let the black community have anything nice. Her words resonated with me. It pierced my heart. I understood where she was coming from. As we start to see the dynamics of our urban environments change, we must make sure that there's a concomitant investment in black and brown communities in the beautification and improving the aesthetic quality of those communities, but also the benefits that the natural environment can offer to those residents and keeping them intact in their neighborhoods. Lastly is the green economy. Our Grown Entrepreneurs Initiative is just one slice of what's possible, offering youth opportunities to work on different projects throughout the city, offers a chance for educational enrichment through a STEM-based focus, but also the opportunity to develop a skill and professional development as well. We need to encourage our youth, who are more likely to be lifelong residents of New Haven, to get involved early and invest in their time and their sweat equity into beautifying and improving the quality in their community, and we should pay them for it on top of that. One of the things that Green Spaces offers is healing. Recently, you may have read in New Haven Independent about the Truman Street Garden, and you may have read about Sierra, a young woman who was fatally shot in the head. Residents came out to the Truman Street Garden and sought respite. They convened, hugged, some cried, some rejoiced in being able to see another day. But they saw that space as a place for healing. Now, there's something called crime prevention through environmental design, and that's utilizing green spaces to address what some criminologists have called the broken windows theory. And the approach to the broken windows theory brought increased law enforcement presence in black and brown communities. And essentially it means if there's blight, if an environment looks like it's not being cared for in an urban setting, then there's an increased likelihood for criminal activity. But what they missed was that you should just fix the broken window, right? So what if we invest in more green spaces and the beautification through horticulture, permaculture, urban agricultural initiatives in our neighborhoods? And if they were strategically placed in areas of high crime and high violence, could we not add to our portfolio of resources focusing on addressing this egregious issue that's been plaguing our communities for far too long? Also, in speaking of PTSD and victims of gun violence, they have a memorial garden, a green space that's dedicated to people who have lost their lives to gun injury. A lot of times we focus on fatal gun injury, and that hits home the hardest. I've lost a number of friends to gun violence. But a larger number are those who survive their non-fatal gun injury. And green spaces can be used to offer therapeutic health benefits to victims of gun violence, stress reduction, getting people to be able to find this place of calming. Because remember, they may have suffered their gun injury in their community and they're going right back to that same community. They're not relocating oftentimes. So we need to improve the conditions in the community so that there can be space for healing. We know that there's a great deal of human ecological value, and we need our municipalities to make the necessary investments, but we need the right kind of professionals. We need landscape architects who have an environmental justice inclination. And as we think about urban design and planning, we need to operate through this lens. We also want to think about the other benefits, like stormwater and flood mitigation, again, reducing heat islands, and also pollution reduction, cleaning the air. We know that the more green that we have, the fresher the air we'll have. We also know that relative to other cities in our state, New Haven has a disproportionately high rate of asthma. Again, it's about investing in our environments. And lastly, we have an opportunity through investing in urban agriculture, urban green spaces, to spur a new generation and more diversity in the environmental justice community. Dr. Dorsetto-Taylor shared that 12% of the leadership in the environmental justice community amongst those organizations are comprised of Black and brown leaders. If we engage people in the natural environment in their own neighborhoods, and we support that with sustainable resources, we are introducing people to an opportunity to become more engaged, not just from a defensive posture, right, to address the issues of brownfields and environmental degradation in communities, which far too often impact Black and brown communities, but also from an offensive standpoint, where people will identify with their contribution on the local level to the global climate justice initiatives. But it starts here. If you want to change the world, you got to start from your corner. Thank you.
So we have time for questions. I believe there's somebody with a mic.
Hi, thank you for a very wonderful speech. Does your organization have any focus on or interaction with the homeless population?
We do, but not in the way that I would like. So what we found are a set of circumstances which I think are in part due to the pandemic and also due to broader issues that spurred displacement. So at some of our nature preserves, there are people, our neighbors, who are shelterless or homeless, and they create or set up encampments, most notably the Pond Lily Preserve in the Westville community of New Haven. We also have had people, neighbors, who are shelterless, homeless, if you will, break into our greenhouses to use that as a place of shelter. So we worked with the City of New Haven with their HOT team, their homeless outreach team. We've partnered with them on a number of occasions to conduct outreach to offer services. We refrain from getting law enforcement involved. That is not the proper remedy for that issue. So we encourage them to take advantage of the services that are offered and available to them through the City of New Haven and also through other community-based resources. Some of the food that we grow, we donate to food banks and other organizations that offer food to those who are in need as well. But that is essentially the extent of our interaction with that community.
Thank you again for your talk this morning. What do you see in the next year or two as your biggest challenges for your organization, and what kind of, do you have one or two primary goals that you're looking to accomplish in the next couple of years?
I would say the biggest challenge is keeping up with demand. We have a waiting list of about 96 people who want to get involved with community gardens. One thing they're not making more of in New Haven is land. So you got to acquire more vacant lots or take on, enter easement agreements to manage vacant lots. But there's another challenge that New Haven has ahead of it, which is affordable housing. And so there are many competing interests, and we're not in that arena to decide what's most important for New Haven, but we're here to be that resource when communities, residents are ready and able to take advantage of this kind of opportunity. But the struggle is that in the existing community gardens, there oftentimes isn't enough space. So a lot of the gardens have garden beds. Some of the gardens practice what's called communal gardens. And so they're able to essentially go in together as a team. They have established work days, and everyone pitches in and gets a portion. I would say somewhat connected to that reality are residents who aren't actively involved with the community garden, which begs the question, what is the purpose and intent of a community garden? So last year, we actually got a few complaints for some of the community gardeners about theft. So they reported that people were stealing their vegetables. Someone took my eggplant, my squash, someone took some kale, So we challenged that and said, is it really theft? It's a community garden. No one is picking a carrot out the ground and just eating it. They're going home and preparing it, which means that there's a need. And so we have to shift the paradigm in how people view community gardens. But I would say the biggest challenge is demand. There's a significant need. We're finding that in the conversations that we're having, either just with residents or with institutions like faith-based organizations, that there's a growing need for people to eat more naturally, eat more whole food diets. And that requires demand. Also, location. There are many neighborhoods who don't have grocery stores where they can get out there and purchase fresh produce. So one of the challenges that lies ahead of us is how can we have a more distributive model and offer more opportunities for people to take advantage of what we can grow in the city of New Haven without having to commute to some of our farm properties. And then lastly, staff. With any merger, you have some people who don't have an appetite for a change management initiative. And so our portfolio of properties and programs expanded, but our staff didn't. So, in fact, I'm actually doing the job of what 2 EDs were doing, right? And so we do need to think beyond being a grassroots organization, right? So there's that level of maturation that we need to embark upon as an organization so that we can actually sustain and evolve our initiatives and have more impact in the city of New Haven.
Thank you very much for your work. I had actually 2 questions for you. One is, do you need to do anything special with the soil in the city to make it viable to grow vegetables and other produce? And then the second was, when I was a young boy, one of my introductions like yours into the natural world, for me it was fishing. And I took that with me until I'm an adult now. Does your organization do anything with fishing either in New Haven, in the lakes of New Haven, or along the shoreline that you talked about to introduce children to that as an avocation?
Not fishing, but sailing. We have had conversations internally about aquacultural initiatives, supporting the marine ecology, but not consuming them or catch and release. I'm a vegan too, so I'm definitely not going to encourage that. But that being said, as far as the soil, we do offer soil amendments. We do soil testing. And for the most part, if gardeners are doing what a gardener would need to do annually or seasonally, then the soil is developing its microbiome, it's developing over time, and it's becoming healthier and richer over time. But that being said, we do provide soil to the community gardeners as well. We test that soil as well, and if we find that it's deficient in certain nutrients or there's an issue with the uptake of nutrients, we'll provide compost or organic fertilizers as well.
Hi, thanks for your talk and for your work, and I want to follow up on what the fellow next to me was just asking. You mentioned earlier about managing vacant lots, and I know that a number of the vacant lots in New Haven have industrial pollution in them. And I just wondered if you are working with any of those things. That's such a huge, huge project, but there's federal funding available to get those things cleaned up. And there's a whole section of New Haven that used to have a lot of industry and manufacturing that has now just vacant lots. I'm thinking, it's kind of hard to describe where it is, but going toward the shoreline and the river down there, you can just see they're just all kind of boarded up. And I wondered if those can be repurposed.
It depends on whether or not there's competing interests, so the city has ultimate jurisdiction and say so. But, I would be excited about those kinds of opportunities. There are state and federal resources for remediation. I'm also interested in exploring indoor growing for a couple of reasons. One, it offers economic opportunities. And 2, you can grow year-round, and you can grow more diversely as well. So when you think about people who are becoming more intently focused on a holistic diet or whole foods diet, you also have to think about medicinal herbs and other plant foods that can be beneficial to communities as well, and some of which we can't grow in New England's environment.
Thanks, Brett. Great presentation. Just a question about where do you go for benchmarks? Is there anyone in the country, any cities, any urban centers that have gotten it right in terms of understanding how to reclaim vacant lots, do the kinds of things that you're doing in New Haven?
There are a few. The exact names are fleeting at the moment, but I know in Providence in Rhode Island, there's a land trust there that actually manages and supports 90 community gardens. There's also right here in our state, in Hartford, there's Knox, there's in Bridgeport, there's GVI, Green Village Initiative, Chicago, Detroit, Oakland, Baltimore, and Atlanta all have initiatives, and of course New York City as well. There's a woman in New York whose name is escaping me at the moment, but she's done tremendous work with not just greening spaces in the Bronx, and I believe in Harlem, but bringing the community and young people in as well. Those benchmarks drive our ambition. To be fully critical of our own initiatives, I personally feel like we got to do better in New Haven. I want to see, I have a vision that I think, you know, I sometimes make my board members tremble, you know, but, you know, when I go through neighborhoods, most recently Newhallville, you know, I nag myself and say, we got to do better, we got to do more, because the demand is there, the expectations are there. And there's a narrative from some that say that folks are indifferent or they're not interested, right? So one might ask, why isn't there a Trader Joe's or Whole Foods in certain neighborhoods? And oftentimes it's a market-based conversation that there's no demand. That's a false narrative, you know? And if they're not gonna bring those resources, if we can't get grocery stores in those neighborhoods, then we need to grow in those neighborhoods.
Great lecture. Thank you. And I look forward, as Dr. White mentioned earlier, the Ag Station here is engaged, I think she's starting in October, a scientist specializing in food security and urban agriculture, first one we've had. And I know our friends at UConn are doing work in extension in urban agriculture. So I would say challenge us to get involved with you. I think I'm sitting here with two of my colleagues from the New Haven Community Foundation and our Ag Station Board of Control. So we've got a lot of collective board members here that I think are ready to be challenged. Secondly, do you do any work with, now it's the School of the Environment at Yale or the Common Ground High School?
Yes, so there's been a long-standing relationship with the preceding organizations now gathered with Common Ground. In fact, currently, we are partnered on a grant, which is the Community Compost and Food Waste Reduction Grant, which is awarded to the City of New Haven from the USDA. And the goal there is to concomitantly reduce food waste that enters the city's solid waste stream while increasing the volume of locally produced compost to support urban agricultural initiatives. So it's a 2-year grant project. In October, we'll start in the 2nd year of that project. There's also a community composting collaborative that's connected to that initiative, which includes a diverse group of stakeholders. But a part of what they are focused on is the equitable distribution of locally produced compost across urban agricultural initiatives in New Haven. And the second part of your question? The Forestry School. Yes, sorry about that. We worked with 2 students during the semester that just passed, the spring semester, and they essentially worked on a community engagement-focused initiative focusing on the Makani Terrace community and the Pond Lily nature preserves. And it's all about essentially focusing on stakeholder engagement and imploring residents to take advantage of that natural resource. There's literally just a chain link fence that separates Makani Terrace and the Pond Lily preserves. But we found that many of the residents don't venture into the preserves. And so we want to make sure that people are taking advantage of it because it's literally right there. But oftentimes people refrain from going into such environments because they don't want to go by themselves or they're concerned about wildlife. And if you do go, you're going to hear things scrambling about. You won't see them, but you'll hear them. But it's safe and it's available. I've also been in several conversations with Professor Gerald Torres, who's with the Yale School of the Environment and the Yale Law School. And they are launching a Center for Environmental Justice. And so they're looking to assign 2 students to work with us throughout the academic year on environmental justice-related projects.
Thank you, Mr. Peterkin, for a wonderful talk. As a director of the Greater New Haven Green Fund, I'm a big fan of both organizations that merged to gather New Haven. We funded probably every project you mentioned and highlighted in your talk. Relating to food issues, gardening is great. I'm a master gardener, but we can only grow food a few months out of the year. And in terms of increasing demand for good food, are you able to work with New Haven schools at all to do educational outreach and teach about nutrition and food issues, through a broader community through the schools?
I think we'd have to look at a model that leverages the existing resources in the school buildings, as there are a lot of schools in New Haven. Some of the schools have community gardens that we support. I would be very excited about such an opportunity. The New Haven Public Schools actually helped us to secure some of the state funding through the State Department of Education for summer expansion programs. They've also had a conversation with us about pursuing the funding opportunity through the Nellie Mae Foundation to focus on environmental justice-related issues. But I do think that there is a meaningful need to have a focus on healthy eating and also growing. And the reason why I think that way is because it's about having an intergenerational impact. So I adopted a plant-based diet, a vegan diet, when I was a teenager. And I come from a Jamaican family. So stewed chicken and rice and peas is a thing, right? So when I came home, I went cold turkey in one day, and I told my mother I'm not eating anything. And she thought I was crazy. But it's been 24 years. So what happened during that course of time was that slowly but surely, pretty much most of my family members adopted a similar diet. In our farm-based wellness program this year, we have a more focused approach to youth. And so the children of the farm-based wellness program, which focuses on adults, because most of them are referred through healthcare providers and physicians, are actually involved as well. And we have a curriculum that's focused on supporting them. And we work with them because a lot of times they're confronted with the means in which their parents are able to afford healthy foods or access it. Sometimes they can afford it, but it's just not proximate, and it's an inconvenience. Also, the way that they approach or receive positive reinforcement. So what's being offered to you as a reward? Is it a cookie or is it kiwis, right? Things of that nature. And so it's about teaching them about the physiological effects of eating healthy, talking about emotional eating, comfort foods, but also quite literally getting their hands in the dirt and they love it. The farms are like a playground for them. The things that some adults might scoff at, they're diving in, you know, arms deep, pulling things out, sometimes killing some plants, but that's okay. We'll grow again. But it's about getting them to sort of connect with the natural environment, to see things coming out of the ground, aside from seeing it in the produce aisle at a supermarket.
Well, I get to ask the last question, I guess. But thanks, Brent, for excellent presentation and all your excellent work. I can see two areas where we can immediately start working together. One, you mentioned the soil testing. We have a soil testing facility, which it seems like you already have for nutritional analysis and things like that. But also our analytical chemistry group can help if there's concerns over heavy metal contamination and things like that, because that's a level of testing that's more difficult to access, so we can certainly help with that. And then, as Terri mentioned, our new scientist, Dr. Lee Whittinghill's coming in first week in October. And actually, one of the areas she's expressed interest in working in is rooftop urban agriculture, which addresses that land issue that you mentioned. So as soon as she's on site, we'll reach back out to you. So
I would appreciate that conversation. Thank you.
Well, thank you again for joining us today. So I have a certificate of appreciation, which is signed by the governor and the Board of Control, and also today.
Thank you so much. Appreciate it.
Thank you for being so attentive this morning. We now have concluded our morning presentations. Please explore the farm, and we'll see you after lunch at about 1:15. And remember to fill out your evaluation form and return it to the registration desk. Thank you. Good afternoon. I hope you all enjoyed your lunch and you're able to visit some of our scientists and scientific staff members, and you're able to appreciate our beautiful Lockwood Farm. We are now prepared to commence the afternoon session presentations for our 111th Plant Science Day. First, I would like to introduce Dr. Walter Krol and Ms. Terri Arsenault. Dr. Walter Krol graduated from the Johns Hopkins University in 1991 and has worked at the Connecticut Agricultural Experiment Station for the past 22 years in the field of pesticide residue chemistry. He developed a catchers-based regulatory program for the analysis of pesticide residues in food. The program was accredited by the A2LA to the ISO standard in 2016 to the newer ISO standard in 2019. He worked with the FDA to co-develop a rapid screen test for PFAS in seafood using catchers with HPLC during the Deepwater Horizon spill in 2010. He worked with the EPA and the University of Connecticut to co-develop and co-validate catchers-based GC triple quad method for analysis of 5 pyrethroid insecticides in lobsters in Long Island Sound. The DAC currently tests hemp grown in the state for THC content on behalf of the Connecticut Department of Agriculture and Regulatory Enforcement. Ms. Terri Arsenault began her work at the Connecticut Agricultural Experiment Station in 1991 in the Department of Analytical Chemistry. She beginning to work in the department, she has developed considerable expertise in the analysis of samples for semi-volatile components using gas chromatograph, coupled with mass selective detector and other conventional detectors such as flame ionization and electron capture. Work focuses on analysis of environmental samples such as water, soil, and vegetation submitted by the Connecticut Department of Energy and Environmental Protection, and produce samples submitted by the Connecticut Department of Consumer Protection for pesticide residues. Additional work includes analysis of food for toxins, beverages for ethanol, pesticide formulations for active ingredients, and solid matrices for PCBs. In 2019, this work was expanded further to the analysis of cannabis samples for total delta-9 on THC on behalf of the Connecticut Department of Agriculture. In addition, Terri has been heavily involved in the development and maintenance of the quality management system, which was required to maintain accreditation ISO standard. So I'd like to introduce to you Dr. Walter Krol and Ms. Terri Arsenault.
Well, I'd like to welcome you all to this fine day out here at the Lockwood Farm. We couldn't have asked for a better day for this event. And I'd like to thank Vicki for the introduction. Today, Terri Arsenault and myself would like to talk to you a little bit about industrial hemp, an emerging crop in Connecticut and for the future. And we're two analytical chemists, and you might ask yourself why we decided to study hemp. Well, in 2018, the Congress of the United States passed a bill, the an industrial farm bill, which legalized hemp growing in the United States. Following this, in 2019, we developed, we came up here and we started planting hemp samples in a field. If you were here in 2019, you might recall we had a hemp plot right over there. This year our hemp plot's moved out a little bit, so it's a little farther out. If you're interested in seeing hemp grown, you can come out to our field and talk to me later after this talk. We had originally grown this hemp because we are a regulatory lab in Connecticut, and we knew once the Farm Bill was passed that we would be involved somehow in a regulatory analysis of hemp. So having this hemp plot really helped us study cannabidiol and THC in hemp. We were supposed to give this presentation last year at Plant Science Day for our 2019 research. However, it got pushed off to this year owing to the COVID situation. So the Department of Analytical Chemistry, that's what DAC stands for, has a long history of testing food samples for various things. We've tested food samples for the Department of Consumer Protection since 1963. And we've tested food samples utilizing gas chromatography and liquid chromatography. Oops. These are some instruments I could just show you. This is liquid chromatograph, gas chromatograph. This testing, not only for the Department of Consumer Protection, but also for the Department of Environmental Protection and the Department of Agriculture, led us into hemp testing. As I said, we got involved in this in 2018. Actually, let me back up here a little bit. So, owing to our analysis in the past for the Department of Consumer Protection, we gained credit. We became a primary lab in the Food Emergency Response Network in 2005. Our work, since we are a regulatory lab, required accreditation owing to the Food Safety Modernization Act passed in 2011. So we started work because we were a member of the FERN. We started work towards accreditation in 2012. And in 2016, we gained accreditation to the ISO 17025 standard and were accredited by A2LA for pesticide residues in food and for arsenic in juice. Since 2016, we further expanded our scope into became accredited for the analysis of aflatoxins in foods. And in 2011, earlier this year, we gained accreditation for the analysis of THC and CBD in foods. in hemp, excuse me. So the Agricultural Improvement Act of 2018, we had to search through that for how our regulatory lab would have to apply accreditation. And there were two big things in that act that were concerning to us. The first one, well, the hemp is defined as a plant of cannabis sativa, and any part of that plant, including seeds, that contain delta-9 THC at a concentration of less than 0.3% on a dry weight basis. This was one of the first things we needed to address in our scope, in our progress to accredit hemp or THC in hemp. The second consideration we had to give was towards what post-decarboxylation meant, because the bill said we need to use a procedure for testing post-decarboxylation of the delta-9 THC. So there were two critical things involved in the testing. The first one was the analysis of THC, and that needed to include a precursor, THCA. And the second one was that dry weight wasn't really defined by the bill. So in method considerations, to get a method to test hemp, we needed to consider several things. The first thing we needed was a quick turnaround time. Something I didn't tell you is we're a regulatory laboratory. Growers bring us samples for the analysis of THC. We turn them around in less than 24 hours because they must harvest it from the field within 15 days. The second thing we needed to do is comply with the delta-9 THC requirement in the Farm Bill. Many of you might not know that when hemp or marijuana, when hemp or cannabis sativa produces THC, the plant itself produces the precursor of THC called THCA. This is converted by heat or other methods to THC. That is the psychoactive ingredient, not THCA. So typically, plant material doesn't contain anything that's psychoactive. It must be converted by heat. So when I mentioned earlier we needed a post-decarboxylation route, any analysis of hemp needs to include THCA and THC. Any reporting that we do needed to, this is all specified in the Farm Bill, must specify either a pass or a fail value. Less than 0.3% means it's hemp. Greater than 0.3% means it's marijuana and not legal. So any decisions that we make about the quantitation of hemp or the quantitation of THC in the sample must include some sort of measurement uncertainty to ensure that that the value is less than 0.3%. Finally, we need some sort of thing for measurement traceability in the method, and that is asking simple questions like, is the lab getting the same results regardless of the analyst or the time? Perhaps the easiest way you can envision measurement uncertainty is if we look at two simple terms, accuracy and precision. Accuracy is how close you are to what the actual value is supposed to be, and precision is how close all of your analysis come to one another. So you can see pretty simply, in a case of low accuracy and low precision, that's a lab we do not want to be, our data can be all over the place. You can also see that you could have very high precision, but you could be missing the target and not getting the right results. Your lab might be very good at producing something that's wrong a lot of the time. What ultimately you would like is a method that can produce high accuracy and high precision, meaning you always get the right answer. So basically, we designed a simple method, a simplified method, by talking to other laboratories and working in our lab. Whereas we obtain a sample of hemp from a grower, and we dry it in an oven at 90 degrees C overnight. The drying process really is just so that it gets dry enough so that we can force it through a sieve to remove stems and seeds. We then take this material that we've sieved and add, using 0.2 grams, we add 25 grams of methanol. This is injected onto a gas chromatograph, and a simple runtime is 10 minutes. And Terri might tell you that yesterday at about 4:30, somebody came in wanting the results, or 2 days ago, somebody came in wanting the results overnight, and she was able to do that for them. Finally, we need quality control in all our analysis. This is one of the most important parts to make you an ISO laboratory. So how we did this and how we get about this is using things called certified reference materials. We run blanks, we run sample duplicates, and we ensure that we test samples in proficiency testing. I've shown you here a control chart. No need to get too involved. But what we do is we take a sample of dried hemp material from a provider having a known value, for example, of 0.193. And every time we run a sample, we run this sample. And we get a result, and a result, and a result, and a result, and it goes, and so on. So even though this kind of looks messy, it actually all centers right on the center line, right where it should be, just a little bit over the average value of 114%. Well, I've told you a little bit about how we get the method. Terri is going to now talk to you about some experiences we had growing hemp for the past 3 years, and she's going to tell you about her experience in analyzing the hemp and some very interesting results. So without further ado, here's Terri.
So after the passage of the Farm Bill in December of 2018, in 2019 we had our first hemp plot, and then in 2020 we'll consider 2019 our learning experience with hemp, because it does take a little bit of learning. If anybody is out there as a grower, they know that. In 2020 we actually really started to get some decent results, and now in 2021 we're expanding that research further. So basically I'm going to concentrate on our 2020 sample plot and what we did and what we learned. In 2020, we grew 8 different varieties of hemp in the plot. We had them all set out in different rows. 6 of those rows were feminized seed. Feminized seed means that the plants only produce female flowers. That is by far the preferable state, because that is where the most THC and CBD is produced, is in the female flowers. The male flowers produce pollen. Pollen produces seed. Seed actually reduces the yield that you will get off of your plants. So you definitely try to exclude those male plants. There's 2 ways of doing that. The first way is to buy feminized seed. The second way would be to actually go out there and clip out the males, which is a lot of manual labor. In 2019, we had non-feminized seed, and we planted that seed again because it did have some unique properties in 2019 that we wanted to further investigate in 2020. But that did mean a lot of extra work of going out there and clipping out the male plants. We started the plants from in the greenhouse from seed. They were planted in the field on June 25th. We did rows that were 5 feet apart, and we planted all the plants about 4 feet apart, because if you wander out there, you'll see that the plants do get quite large. We used black plastic to control the weeds, and we have a drip irrigation hose underneath the black plastic to make watering easier. This was what we found to be the best way to grow hemp, but obviously there's many people out there who are simply going to put the seeds out there, and it will grow. I suppose it is sort of a weed. On June 25th of 2020, you can see planting day there, and you can see our seed tray sitting in the front. That's what it looked like after about a month in the greenhouse. And then on July 24th, you can see that those plants are maybe 2 or 3 feet tall, mostly doing well. There was some occasional plants that didn't, what we'll call failure to thrive, but by and large, everything was growing pretty well. August 26th, things are really coming along nicely. The plants are 4 to 5 feet tall. And on September 9th is when you can start to notice that some of the varieties are really getting ready or getting fully mature at that point. The female flowers start to come out probably in another week or 2, but really it takes maybe about a month before they have a maximal amount of CBD. By September 16th, the plants, they don't really get a lot taller at this point, but they can continue to exude the sticky material on the buds. That sticky material from the female flowers is what's going to capture the pollen if we had pollen, right? But we try to keep the pollen out, which means that the female flowers will continue to exude that sticky substance, which contains the CBD, which is the thing that you want as a grower. Around September 30th, you can see that some of the plants might be starting to turn yellow. They're a little bit past their peak. We noticed a little bit of difference in some of the varieties, and it's really hard to see from this picture, but if you could walk down those individual rows, you could see that while some rows were well past their peak, some of the other rows were just really hitting their peak around that time. Challenges in 2020, we had Storm Isaias, which came blowing through here August 6th, which if we had had Plant Science Day, I think it would have been the day of or the day after, something like that. The day before, which meant that Plant Science Day would have been a little bit of a nightmare. So when we were diverting to the indoor Plant Science Day, as much as a disappointment of that was, clearly if Storm Isaias had come through that night with these tents, we would have had a problem. And one of the problems that we had is that the wind comes whipping down this hill and across the field. And we had some significant wind damage to the plants. So being an analytical chemist, I actually don't grow the plants. Thank you, Rich Ciccarelli, for taking good care of them all year. And I came out after the storm and I said, hey, I don't understand. Why are the edges of all my leaves brown? And he said, well, is it on that side? And I said, yeah. And he was like, oh, it's wind damage. So it literally dries out the leaves, and you could tell because of the way that the damage ran across the plot. We also noticed that some of the varieties, and you can see it here, I get the stronger pointer, I don't know, oh, the bottom. Okay, this plant right here, some of the varieties were very tall and straight. This variety totally laid down during the storm. So over here you can see they're laying down on the ground. Some of the varieties that were nice and bushy, they had less effects from the storm. So there is quite a bit of difference in the way that those varieties look. I know that there was some growers out there, I feel very badly for you, they ran around and they actually had to stake up their plants last year after this storm following this damage. Because we never really intended to harvest these plants, because all we're doing is analytical testing on them. We did not stake them up, but again, that would have been a significant amount of input of effort and labor for those poor growers. I don't necessarily feel your pain, but I'm sure that others do feel your pain. Last year, we had some pretty significant damage from corn earworm. I don't know if you can see this very clearly here, but these buds are brown, brown, brown. I went out on September 9th, and again, as an analytical chemist, I've been in this field every week, but I had not noticed that there was corn earworm. And by September 9th, I went down, and all of the plants that were ripe in some of the varieties, literally the flowers, which is where all your CBD is that you want, were brown and dead. And I was horrified, and I went to the farm manager and I'm like, I don't know what to do. And he fortunately does know what to do, and he sprayed this with Pyganic, which is an organic pesticide to get rid of some of the corn earworm and save some of this crop. There was some minimal damage from the corn borer, but the corn borer was not nearly as bad as this corn earworm because it literally attacks the crop that you want, which is the fully mature flower. So now, here's a summary of our growing experiences. We definitely in 2019 found that removing males is a lot of work, and you must check your plants every few days. Those buds, once they develop the pollen very, very quickly. And what we found was that that was a really lot of work, and we decided the following year to use only feminized seed. Highly recommend feminized seed. Hemp varieties are exceptionally variable in the way they look when you walk out through the field. I had some people who are plant horticulturalists, and they were like, yeah, those plants don't even look like the same variety. They are the same variety. They grow right next to each other. Some of them have skinny leaves. Some of them have fat leaves. Some of them have like veins that are visible, and other ones look more shiny, and they're all supposed to be the same variety. It did lead us to believe that the hemp seed varieties are really not very well developed at this time. That's not entirely unexpected. The Farm Bill was only passed in December of 2018, and that year in 2019, a lot of the seed producers were probably having some trouble producing seed or getting seed out there fast enough for all the people that wanted to grow hemp, and the seed is very expensive. As I already mentioned, there was significant damage from insects, there was wind damage. Sometimes you look through the plot and there's just stunted little plants and they never grew. I was told by some of the plant pathologists around here that there's probably some sort of root rot that create a failure to thrive situation. In addition, if you're thinking of becoming a hemp grower, just keep in mind that this has a very strong odor. When these flowers start to come out, which will be within the next few weeks, people that drive onto the farm, and I mean down by the lower barns, can smell this, especially in the morning when the wind is coming whipping down. It's something to consider if you like your neighbors and your neighbors don't like the smell. Not everybody does. Some do, some do not. Also, this crop is very sticky. Every time I walk through it, I'm like covered in goo, and you gotta like wash up to your elbows. That can be very difficult in some ways to work with. This is the part that I'm most familiar with because as an analytical chemist, this is what I did. I went out and I collected samples from all 8 of those varieties every week. I went out on Wednesday mornings, usually around 9 o'clock, and I snipped 1 2 to 3 inch piece of every single plant that constituted that variety and took it back to the lab and tested it for total delta-9 THC and total CBD using gas chromatography with flame ionization detection. For all you people, some know what that is. If you don't and you want to know it, I can explain it to you later. In addition to that testing of the varieties, we also tested some select individual plants. That was just to see what goes on with THC and CBD within individual plants and not just the whole row of plants. In addition, on 3 separate occasions, I tested every single plant of those 3 varieties on a single day so that we could see at that given time how much THC each of those plants contained. These are some of the results. Now, I did 8 varieties, so I had 8 graphs, but this is only a 20-minute talk, so I'm just going to present 3 of them right here. And you can see that in this graph, the total CBD concentration range is on the left over here, right? And that graph goes from 0 to 20%. And on the right-hand side, we have total delta-9 THC. That graph goes from 0% to 1%. So they're different axes, but I just wanted to show you how THC and CBD basically have a similar pattern in the variety, but they're at dramatically different concentrations because this is considered hemp. So this starts on August 12th, and we sampled all the way until October 28th, every single week, as I mentioned. And what these graphs are showing us is how quickly CBD and THC will escalate in these plants. Walt had mentioned earlier that the growers get 15 days from the initial test sample that they clip off of those plants to complete their harvest. Within that 15 days, you can see that a plant can go easily from here to here, which is at least maybe a doubling in concentration. So that 15 days is, it's very crucial that growers be very careful about how long they wait from the timing of that harvest before they finish their harvest, because the THC content can escalate very, very quickly. In this variety over here, this Young Sim 10 Lot 17 variety, the THC and the CBD don't really track very well together. One of the things I knew about this plant from 2019 was that it was a little bit unusual, and the reason why I sampled every single plant in this row was because we were trying to figure out how come these plants would not track closer, because everybody tells you that they're supposed to track together. Okay. So this is where I went out and collected every single plant, and on September 23rd, I collected the Young Sim 10 lot. There is a lot of variability between plants of this variety. And what I want to point out on this graph is that the axis over here is the total concentration. The blue bars represent the concentration of the CBD, and the orange bars represent the concentration of the THC. Some of the plants in this variety have an almost equal concentration of THC and CBD. This is not really supposed to happen with a variety that is considered hemp. In hemp, the ratio of CBD to THC should be about 25:1, which you can see it is for most of these plants, but not for all of these plants. In fact, 4 of these plants had an almost equal ratio. What's particularly concerning to me is this plant right here, which has 7.5% THC. That is clearly a psychoactive level of THC. There is, and also I should point out that for some reason, this plant, number 11, has an almost equal ratio of THC to CBD, but it doesn't really have a lot of either one. And if you're a grower, this can be like very concerning, because you have all these plants and you think you're going to get 8-9% CBD out of them, but you can have these really poor performing plants within that variety. This variety was much more consistent. I had 33 plants out of here, and on October 14th, I tested every single one of those 33 plants, and one of those plants out of this variety had a psychoactive level of THC at over 8%. The other 32 of those plants were perfectly fine, and the ratio was correct according to what you would expect. In addition, this plant, up to 21%. So some of these plants had a really good production of CBD, but again, there's some plants in there that really don't. And you can see this one right here just has an overall low level of CBD production. The last variety that I'll talk about where I tested every single plant is the Cherry Abacus 2.0. It was collected on October 22nd, and this variety had no high THC plants within the variety, which is good because it really shouldn't have that. But as you can see, we go from about 15% CBD to about 23% CBD, even though all of these plants are theoretically at the same variety. So now I'll just talk a little bit about some of the plants that I sampled individually. On August 12th, I went out, and in the Abacus 2.0 plot, what I found was that there's plants growing right next to each other, and one of those plants has flowers. It's maturing. The plant right next to it, 0 flowers. So I tested those 2 plants individually every week, starting on August 12th and going until October 20th. At that initial sampling, the THC content in the plants that had no buds was 0.09%, and it had 2.1% CBD, which would be an expected ratio. The plant with buds had almost double the amount of both THC and CBD, which again would be expected because we know that the flowers are what produce the THC and the CBD. As we tracked those plants individually across the growing season, what we found was, even though the plant that was initially less mature, which I'll point to right here, started out at the lower rate, it actually ended up having more CBD and THC by the time the growing season was over. I will also say that if you look at these curves side by side, oops, did I lose it? It's almost virtually identical. In other words, the CBD and the THC will overlay each other almost identically in the plant. So this does show that THC and CBD increase together at a specified ratio for the individual plants. But early maturity does not equal more CBD production. This way? Oops, wrong way, sorry. Okay, in conclusion, gas chromatography with flame ionization detection was a really robust method of analysis. We are able to turn results over for most people within 24 to 48 hours. Any growers in the audience, hopefully you've been happy with our turnaround time, because I know that one of the big considerations in the Farm Bill was that they said that labs were having a difficult time turning samples around quickly enough to allow that 15-day harvest window. The new regulations actually do allow a 30-day harvest window, primarily because of the issue with the turnaround time. Our concern continues to be that because of that 30-day time window that USDA just implemented, that some of these CBD products will have an excessive amount of THC. So we would still encourage growers to stop or to harvest those samples as soon, or harvest their crop as soon as they collect, as soon as they can, so that they don't have any issues with excessive amounts of THC. CBD and THC do spike around the beginning of September. Individual plants do mature at different times. And at full maturity, there is a high likelihood that a number of these varieties will exceed the 0.3% THC limit. In general, hemp varieties appear to have higher variability and poor predictability than other plant varieties. You know, if you get a cherry tomato, it does have cherry tomatoes invariably, but this is almost like getting a beefsteak tomato in the middle of your cherry tomatoes, and you would be like, what the heck is that? Okay. I went the wrong way again. Sorry. Okay, so I would like to acknowledge Dr. Jason White, who has always supported this, my current department head, who has continued to support this, Rich for obviously doing all of the growing, Mike Amarata and Kitty, who both helped me in the laboratory with processing these samples, because in addition to doing these samples, we also have the regulatory samples coming in from growers, so without them, never could have handled all of this, and of course the entire rest of the staff of the Analytical Chemistry Department. And now, if you have any questions, did we get it in 20 minutes? Good. Did I turn that on or off? Oops. Okay. Any questions? Yes.
Okay, sorry. Hi, thank you for your presentation. That was really enlightening, especially how you went through the trials and the data that you got from that. My question for you is whether you have encountered either in the literature or your own understanding of the botanical function of THC, given that there's an emphasis on its psychoactive component after the fact, but what role does it play botanically, and why might we see like spikes in THC in relation to the maturity of the plant?
I honestly don't know the answer to that. I mean, I've heard a lot of stuff about possibly making it insect resistant, you know, things like that, but I really couldn't answer that question. Sorry. 3 strikes and I'm out, right? I think back there, yeah.
Hi, Terri. I just had a quick question on your outdoor garden plots. It looks like you were using black plastic. Is there a better alternative since our world's drowning in plastic?
You know, obviously, if you go out, there's lots of ways to control weeds without using black plastic. You know, you can. I don't grow. Vicki could probably answer this question more, but I mean, you know, mowing, mechanical weeding, things like that, yes. They do black plastic because I think we have a black plastic machine that lays it down, so it reduces our labor costs a lot. So it can be beneficial for that purpose.
Okay, thank you so much.
Thank you for the delightful talk.
I wonder about variability within one plant and what the source of that variability might be. Thank you.
So the variability is like between plants of the same variety, and I think at this point the suggestion is that the genetics are just simply not well developed enough. You have to do multiple back crosses to really get true breeding varieties, and because this is a relatively new field, they just simply haven't developed the varieties enough. Good? Thank you all.
So our final presentation for the day, I would like to introduce Dr. Kirby Stafford. Dr. Kirby Stafford is the Chief Scientist, Head of the Department of Entomology, and the State Entomologist Plant Regulatory Official at the Connecticut Agricultural Experiment Station. He joined the Experiment Station in 1987 to start a research program on the ecology, distribution, and control of the black-legged tick, Ixodes scapularis, with a major focus on natural, biological, and integrated tick control. And more recently, the Lone Star tick, Amblyona americanum, a newly emerging tick species in Connecticut. Dr. Stafford has authored or co-authored 86 articles in peer-reviewed scientific journals, chapters on tick management in 2 books, and produced the Tick Management Handbook, which is our CAS Bulletin 1010, and the Fly Management Handbook, which is our Bulletin 1013. Presented research at national and international meetings, serves on multiple regional and national tick-related committees, and given over 1,300 media interviews and public talks. Dr. Stafford oversees the Office of the State Entomologist and works closely with the USDA APHIS PPQ, U.S. Forest Service, and deep forestry and forest health-related issues. Prior to coming to Connecticut, Dr. Stafford worked at Penn State. Dr. Stafford attained his PhD in medical veterinary entomology from Texas A&M in 1985. He received a Master's of Science degree in entomology at Kansas State University and a Bachelor of Science degree in entomology at Colorado State. So, Dr. Stafford?
Thank you, Vicki. Good afternoon, everyone. We're just going to wrap up the presentations here on a little bit lighter note. Obviously, I hope everyone's having a good time because this subject today is, of course, Plant Science Day celebration, a history. And so what I'm going to do is basically give you a history of how a Plant Science Day came to be. I'm going to highlight some of the past Plant Science Days with some historical pictures, which I think you will find quite interesting. I'm going to kind of review some of the upgrades and changes that have happened here in our field days over the years, and wrap it up with basically what you see here today. So just to remind everyone that the experiment station itself was the first one in the United States, and we were established by the General Assembly in 1875. We moved to our New Haven location in 1882, and as you can see in the picture here are some early station plantings roughly around 1910. Now, another adjunct to those limited plots at our original New Haven site was the Centerville Farm. And this was located what is now, for those of you that may be familiar with Hamden, Connecticut, is where Norwood Avenue intersects with Whitney and Washington Avenues there in Hamden. And at that farm, there was experimental breeding work was done on corn, tobacco, testing lime sulfur, summer sprays for fruit trees, and a handling of a number of other field crops. There are also plots available at various cooperating farms. There was one, for example, in the Spring Glen area of Hamden, which is now a shopping center. But all of these sites were basically leased in 1908, 1909, and 1910, and they were easily reached from the New Haven campus by trolley. Now, our first field meeting was held on the 10th of August of 1910. So there was an informal gathering at our New Haven location to dedicate our new building, the Johnson Laboratory. And then afterwards, about 400 farmers and their wives, according to this quote, were present. And then in the afternoon, they went from the station to that Centerville field to inspect and discuss the research that was being done there. And at that time, it was intended to hold this summer meeting each year. And here is our classic photograph of people gathered for that first field day for the experiment station. I just love this shot. Look at the kind of clothes that people are wearing. And if you look really carefully, you can see the trolley tracks there along the bottom of the picture. Now, on November 28th of 1910, the station bought, that is the Board of Control, bought a farm of about 20 acres at Mount Carmel here for about $6,000 with monies provided by the Lockwood Trust, which was established in 1896, hence the name of the farm, the Lockwood Farm. Now, this site had not been cultivated for quite a few years, and as noted in some of our literature, the west end was overgrown with boppin, birch, sumac, sweet fern, and blackberry vines. So the station set out a new orchard of apples and peaches for experimental work, and then began experiments on the effect on the crop and on the soil of various fertilizers and manures, and another on the handling. There was actually an old and more neglected orchard here at the site at the time. And that original center field was given up in the spring of 1912. Now, this is a map of Lockwood Farm, and this shows you basically where the original site of the 19.25 acres was purchased. So it's that area of the farm where you all drove in down by the lower barns. Here was a field day in 1914. At that year, a barn was built on the site of an old ruined barn at a cost of $1,593.29. And a second field meeting was held at this Mount Carmel farm here that August with about 250 people in attendance. Then in 1915, an additional 16 acres was purchased and added to the farm. And then in 1928, an additional 4 acres was purchased to add as the farm slowly began to expand. I love this picture. Here's a shot of field day here at Lockwood Farm. It's an undated photo, but approximately somewhere around 1915. I just love the picture of the old cars here gathered out in the field. So how many are all parked out in the field today? All right, okay. Well, just think about the people coming here in 1915, and there are all their cars all lined up there and parked. Here's another shot somewhere around that same time. You can kind of sort of see Sleeping Giant there in the background, the large tent set up, and again, you can see the cars just barely kind of, and people gathered around there under the tent. Now, one of the big accomplishments here at the farm was the discovery of hybrid corn. Now, Dr. Donald Jones was hired in 1915. And against genetic dogma, he conducted trials crossing 2 crosses of corn. He found that this double cross vastly increased yield. And he published this method in 1919, and the station did not patent it. So it was available to basically for all the farmers. And this shot picture of him is basically from a field day in August of 1931 in which Donald Jones was honored for his accomplishments. Following the time sequence here, you can see what the field day looked like in 1924. Again, the large tent. You can see the older, vehicles of the time there. And then subsequently here under that same tent, you can see people gathered around and gathered to listen to a presentation at the time. Now, interestingly, there was no field day in 1925. The Board of Control voted to admit the usual field day because an exhibit was going to be sent to the Charter Oak Fair and because the station celebrated its 50th anniversary on October 12th of that year. And you can see the brochure there in the image. Here is our field day brochure from 1940, and Dr. Slate was the director at the time, and I thought I would just read this part here. On August 21st of 1940, while the station farm and laboratories are open to visitors throughout the year, this day is a special at-home to all citizens of the state. We extend to you a most cordial welcome. Mount Carmel Farm is our principal outdoor laboratory where many of our experiments with plants are carried on. There are also the tobacco and vegetable farm at Windsor and the experimental forest at Rainbow, which no longer we have. This book gives directions to finding the various plots, and you can also find staff that are there to explain. Some things haven't changed, folks. We're still doing that here for you all today. And certainly welcome you here to the farm as always. Here's a picture of a plot with visitors there, and again in 1940. And another shot of the tent in 1940, which at this point was located down by our lower barns, nearby right where you drove in. Now, there was no field day during the war years of 1942 and 1944. We had 16 staff that were serving in the armed forces. So after 3 years without a field day, attendance exceeded 1,000 people in 1945 and again in 1946. No mention is made of field day in 1950, which is when we celebrated the station's 75th anniversary in New Haven in that September. There's actually a few other years when it's possible there was no field day. At least there's no mention in our records, so I'm really unclear whether a field day was actually held on those few specific years. Here's another shot of field day in 1946. Again, the big tent down by the lower barns. And then in 1948, the Board of Control purchased another 14.5 acres, continuing to expand the size of the Lockwood farm. Here's a shot of field day in 1949. And here's a shot of people gathered underneath the tent there in 1949. And another picture of farmers gathered around looking at one of the plots here, again, August 24th of 1949. And here, where we're seeing coffee being served to visitors here in 1949 as well. Now, one of the new innovations for 1949 was a tractor-trailer tour, which went on for a number of years. That is subsequently was replaced by the bus. We're not having the bus tours this year due to obvious considerations due to COVID-19 and social distancing. Now, the barns that you see up here, these are upper barns here, were built in 1948 and 1953. This is a shot of the barns and sleeping giant in the background that was taken in 1961. And the farm continued to expand. So in 1985, another 16.7 acres at the far end of the farm was purchased, and then a subsequent 2 acres where a cottage is located, just actually kind of back behind us, was purchased in 1997. Here's a shot of our Plant Science Day in 1988. This is Dr. McClure giving a lecture on the hemlock woolly adelgid, and at the time, underneath the tent, we were using posters for our presentations. We still had tours, as you can see in this site here, that and a sign located by our barns. Walking tours start here in the various times that are given. And here is another shot of Plant Science Day in 1988 underneath the tent, which was actually located right in this locality here, actually a little further back. And then again, we also, at that time, had barn exhibits, which I hope all of you managed to catch here today up at our barns up there. Now, at this point, things begin to change and evolve. The garden, and I encourage, if you haven't seen it yet, I do encourage you to go over and see our bird and butterfly garden. That was begun in the fall of 1996 in a partnership between the Experiment Station and the Federated Garden Clubs of Connecticut. It actually really is a very lovely spot, particularly in the early morning hours. And then the 1920s cottage, and if you look up in the upper corner, you can see what that cottage originally looked like. It was actually a Sears cottage, and it was, it was purchased by the Board of Control in the 2 acres adjacent to it here in 1997. It was extensively renovated. Now, at this point, that all of this brought the farm up to just a shade over 75 acres, which is where it stands today. So other innovations we added our technical demonstrations in addition to the main lectures here that were under the tent. There was an old insectary building that was demolished and replaced with another insectary back in 2001. And then beginning in 2002, we began inviting outside groups to establish tables, which at the time were set up in what I will call the avenue here in this little corridor off to my left. And as you can see here, this shot's from 2008, and you can see the tents kind of all aligning that just off to the side from the main tent at the time. As we continue to innovate and expand our program to reach more people, we began the Children's Passport program, the Kids Corner, inviting Girl Scouts. As part of that, you may have seen the Kids Corner booth up here along the roadway. And we also have these unique, very beautiful patches, which were designed by Catherine Dugas, that are available for those that participate in the Passport program. And the children can go around with this passport to designated stations and get their passport stamped. Here's just a general shot of a picture of Plant Science Day in 2007, very much like what you're seeing here today. And the tick tent, which is just over there behind us, was actually located down by the barns, but in 1997 it became more broadly the public health tent as we consolidated our tick, mosquito, and wildlife programs underneath that one tent. And here's a picture of some visitors here on Plant Science Day in 2008, just visiting one of the mini plots here at the station. Now, there are 5 governors that have made an appearance to speak or remarks at Lockwood Farm on Plant Science Day. And of course, today we also had our Lieutenant Governor here briefly. But these include Governor Daniel Malloy in 2014, Governor Lowell Weicker in 1991, Governor Ella Grasso in 1975, Governor John Dempsey in 1960, and Governor Wilbur Cross in 1931 and in 1934. Now, the pavilion that you're in here today was constructed by the Board of Control in 2015, so its first use was for Plant Science Day in 2016. And for those of you that are curious, the silo cell tower that you see right here was constructed in 1998. What you're seeing today, we added, remember that slide picture I showed you of Dr. McClure standing at that poster? Well, obviously, we've come a long way since then. We're now doing PowerPoint presentations. We actually have 2 large screens. We've added the additional screens that you see for those of you that toward the back to be able to better see the presentations. And I'll just add in some trivia here. As I already previously mentioned, there was no field day in 1950 during the war years of 1942 and 1944, and possibly a few other years. The only Saturday field day was in 1958. The Mount Carmel Field Day was first called Lockwood Farm Field Day in 1960, and for another 8 years after that was called the Science at Work Day. It was first called Plant Science Day in 1969. Now, between 1960 and 2018, the average attendance was about 993. And 973 people attended in 2019. How many people attended in 2020? We had a virtual Plant Science Day simply because obviously we could not be open at that time. Attendance has actually exceeded 1,500 on 3 occasions. This was in 1934 with Governor Cross, 1961 with Governor Dempsey, and in 2002 when Roger B. Swain delivered the Samuel W. Johnson Memorial Lecture. It was a wonderful day, but I must say a very busy one. So you think about, as I've kind of highlighted a little bit, the improvements and changes over time. We've added the bird and butterfly garden. The main tent here has been replaced by this lovely pavilion. We switched from posters to PowerPoint short talks. We've added multiple screens. We have a new sound system. We've improved the registration process. We added the technical demonstration tent talks on, real fundamental things that could be like beekeeping or how, container gardening and things like that. We have the new cottage that I mentioned that was renovated. We started and tried to increase the involvement of children with the Passport program and Girl Scouts. And we, of course, expanded with the participation of outside exhibitors. So there's a lot to see and do here at Lockwood Farm here on Plant Science Day. So I'll wrap up here. Field Day to Plant Science Day. It's been 111 years and counting. It's a beautiful location. I hope you all have a really, really great time. And if you have any other additional questions or follow-up, here's my contact information, and I'll take any questions people have at this time. Thank you.
Thank you, and it's nice to hear about the changes. Would you highlight some of the technical achievements that have been done by the station, plus what's been demonstrated here from the field trials? What has come out that's lasted? What has come out that's been a flop?
Okay, well, that will be here. I'm not sure I could bring up, the diversity of the plots and research that's been done here at the farm is immense. I'm not sure I could remember or cover all of those. The one advantage when we had the bus tours that allowed people to go around and easily see all of the plots and that's going on right at the moment. Given the 20 minutes here, I only highlighted the one discovery of hybrid corn at the time. But yeah, afterwards, you were welcome to talk to me about some of the other efforts and work that has been done here.
Is it on your website?
And there's a lot of information available on our website as well. Any other questions?
Can you tell us a little bit about that hybrid corn?
Okay, so it was a double cross, and basically the idea at the time was, what would that do? And he discovered it just increased yield. And Dr. Jones did not patent it, and it was picked up by other companies and produced. And so for a long time, that was where if you looked at yield of corn in the United States, you basically took a big jump at that time. And then, of course, then subsequent to that, there's been a lot more genetic work done on increasing yield in corn beyond that double, the hybrid corn that he developed. But that was a real innovation of the time and really jumped up national corn production.
Thank you.
Any other questions? If not, thank you very much and enjoy the rest of the afternoon.
Thank you. I would like to thank you all for attending our 111th Plant Science Day. Please enjoy the remainder of your day and drive back safely to your families. Please remember to fill out our evaluation form and return to the registration desk. We really appreciate your feedback. So see you next year. Have a lovely afternoon, and be sure to visit a local farm and support Connecticut agriculture. Thank you.